from the WHO Emergency Program, and it's my pleasure to welcome all of you today. Number 2, please do not use the chat service to say hello to other colleagues. Please use the chat to raise questions, comments on the topics of today. And then finally, we are very honored today because the chairperson of this consultation is going to be Professor Stanley Plotkin from the University of Pennsylvania, who almost doesn't need an introduction and is a pioneer in vaccine development and evaluation. Professor Plotkin, as you know, not only has invented vaccines, but has helped us to understand how to define correlates of protection. So, Stanley, thank you very much for agreeing to be our chairperson today. The floor is yours. Is it only me, or can the others hear Stanley?
Sorry. I'm sorry. What I was saying was that I think all of us have been impressed by the variability of the SARS-2 Sarbecovirus and the question of how we can control its many variations. And so the subject of this meeting is what to do about that variation and how to develop vaccines that will be broadly effective. The next slide. The fact of the matter is that coronaviruses are ubiquitous, particularly in Southeast Asia. So this map simply identifies many of the coronaviruses that have been isolated, largely from bats, but also coronaviruses passed from bats to small mammals. And it is said, based on the work of Marc Eloy at the Pasteur, that a virus from Laos actually is the closest in sequences to the virus that has escaped into human circulation, SARS-2. Next slide. As you all know, the structure of the beta coronaviruses is quite similar, as you can see from this comparison of SARS-1 and SARS-2, with the importance of the external spike protein, but also other proteins, and I hope we get into this discussion during the meeting, whether other proteins may be useful as vaccine antigens. But of course, for the moment, the spike protein is what most people are using. Next slide. The other, another key point about these viruses is their variation. And this is a probably incomplete list. It doesn't have Omicron on it, but it shows that the genetic variation of beta coronaviruses is considerable and continues to occur through 2020, 2021, and into 2022. Next slide. And specifically, variants of concern that are causing considerable disease are listed here. And my point is simply that the variation is not simply a virologic phenomenon. It has an important medical effect on the type of disease and the incidence of disease occurring with these variants. And of course, you all know that the latest Omicron variant is causing problems throughout the world and is continuing to mutate, continuing to develop subvariants such as the BA2 variant. Next slide. Now, our vaccines have, in general, been quite successful. But if, for example, if you look at the action of the Moderna messenger RNA vaccine against Wuhan, Delta, and Omicron, you can see that the Omicron variant is not well protected by vaccines made against the original virus. Fortunately, as one can see on the right side of the slide, booster vaccinations, or really I prefer to simply to call them third doses, do help against the variants, as shown, for example, that many people do develop antibodies against Omicron, but not everybody, and so that is still a problem. Next slide. But I do want to concede that the third doses of vaccines, if they are given, something which is well known in immunology, that is the requirement to space inactivated vaccines. What we see here is the pink lines are antibodies against the Omicron virus, and what we see is that the third dose does in fact raise antibodies, so that those third doses are essential to be given for medical and immunologic reasons. Next slide. So why do we need a Pan-Sarbecovirus vaccine? Well, variants of SARS-2 beta coronavirus may continue to escape partially or wholly neutralizing antibodies induced by vaccines against prior variants. No doubt we can make vaccines against new variants, but that takes at least months, and therefore people will become ill in that time. The reservoir of beta coronaviruses in bats is large, and new crossovers to humans is likely. And if we prepare now, the time required for large-scale manufacture will be reduced, and therefore vaccine can be distributed. Sorry that I'm being automatically muted for some reason. If we prepare now, the time required for large-scale vaccine manufacture will be reduced and lives will be saved. So what can be done? We will hear the answer to that question today because many people have recognized this issue and have started to prepare candidate vaccines. So far, as far as I know, they've only been tested in mice, but next slide actually is a slide from Daniel Culp at my university, the University of Pennsylvania, and who is, as many others, trying to develop a nanoparticle vaccine that will be broadly protective against various types. And if you look at the lower left-hand corner of this slide, you can see that immunogenicity of this nanoparticle vaccine is good against a variety of SARS-2 strains, including the beta as well as the P1 variant from Brazil, etc. And the lower graph, the lower left corner, shows that mice given that vaccine are protected against challenges with various variants. Next slide. But fortunately, we have many different strategies to develop vaccines against coronaviruses. And obviously, they include all of the ones on this slide. All of these strategies have been used and can be used to make future vaccines. I think from what I've heard thus far that the nanoparticle approach has been most commonly used, but that does not mean that we cannot try other strategies that have been successful in other situations.
Stanley, you are mute again. Stanley, you are muted again.
Next slide. So the technology to make Pan-Sarbecovirus vaccines appears to be available. A supply of such a vaccine might be able to stifle a small outbreak of a new coronavirus. Once developed, shown to be safe and immunogenic, a large amount of a new vaccine can be manufactured quickly and used to deal with the adaptation of a new beta coronavirus to humans. And my last slide. We need, in summary, we need a Pan-Sarbecovirus vaccine because coronaviruses are numerous and mutate rapidly. They easily infect humans as well as many other animals. Multiple coronaviruses have already infected humans. New coronaviruses are likely to emerge. Other antigens than the spike, such as the nuclear protein, in my view, should be incorporated, not just as an inactivated antigen, but as an antigen that can be fully expressed and induce T-cell responses. And lastly, a Pan-Sarbecovirus vaccine is likely to work based on studies already done in several labs and a program developed at CEPI to do that. So let's hear from all of you what is being done and what can be done in the future. Thank you. So let us then move on. I would ask the next speaker, Ralph Barrick from the University of North Carolina, the Dean of Coronavirologists, to give us the first scientific talk.
Thank you, Stan. It's a pleasure to be here and to provide sort of this high-level update on major challenges associated with the development of a pan-coronavirus vaccine. I'm sorry I don't have control of any pointer. I'll just note that the immunologic target that's been defined as a protective correlate is neutralizing antibodies against the spike. However, when we consider moving into pan-coronavirus vaccine development, we definitely need to consider the importance of T-cell immunity to conserved regions in the virus genome as well. I'll note my collaborations and consultations with various companies in the lower right-hand side of the slide. Next slide. Well, there are a large number of potential challenges associated with developing a pan-coronavirus vaccine. And they fall into various categories, the first being virologic. At this point, we still have a unclear knowledge of the phylogenetic and antigenic breadth of coronaviruses, especially those that circulate in natural reservoirs like bats. If you look at the right-hand side of the slide, there's lots of coronaviruses that live here that have not been well characterized. We know that SARS coronavirus 2, as well as the human coronaviruses, probably undergo some form of antigenic drift, which has not been well characterized, but is certainly just being recognized to date. To develop pan-coronavirus challenge vaccines, you clearly need challenge models and broad divergent panels of live viruses to begin to evaluate vaccine performance in vivo. And we have to have access to a large number of pan-coronavirus immunologic assays, identification of conserved epitopes, and the consideration of different vaccine design platforms for solving the problem. In terms of animal model development, we need high throughput animal models like mice and hamsters, I think, which are quite robust for SARS, but their utility across the coronavirus family has been less well evaluated, as well as large animal models. It would be nice if some of these models could develop acute respiratory distress syndrome, which is the typical disease, as far as we can tell, that is associated with new emerging coronavirus occurrence in human populations. And it would be very nice to have at least one model of vaccine-associated enhanced respiratory disease that can be used as a control to evaluate in parallel with the best platforms that are moving forward in human clinical testing, so that we know that they're safe. Another consideration is the development of human challenge models for not only SARS-2, but also the contemporary human coronaviruses, which could potentially provide some definitive information about vaccine performance. Next slide, please. And then there's a variety of immune considerations, not only involved in testing, but also in clinical, in small animal models, but also testing in humans. And that's the effect of pre-immune status on pan-coronavirus vaccine performance in naive and vulnerable populations like children. So the vaccine is going to be used in multiple different populations that could affect immune kinetics, breadth, durability, and memory. And better defined correlates of protection that not only include neutralizing antibodies, but T and B cell based correlates, and the potential for antibody-mediated, FC-mediated protective immunity, and how they all work together to provide cross-protection against strains that are included in the cocktails, but also strains that are not present in the cocktail. And finally, a well-defined product development plan, I think, will be critical. Next slide. Now, when you consider, looking at the right-hand side of the slide, when you consider pan-coronavirus vaccine development, you actually have several strategies that can be undertaken. Looking at the broadest level is the pan-coronavirus vaccine that would cover all 4 major genus within the coronavirus family, including the alpha, beta, gamma, and delta coronaviruses. But we also have the capability of developing more focused pan-genus specific coronavirus vaccines. This would be against all alphas, betas, gammas, and deltas independently, which could be then multiplexed or used independently in an outbreak setting. And then we have strategies where subgenus or subgroup specific vaccines can be developed against, let's say, all Sarbecoviruses or all MERS-related strains. And finally, you can, for SARS itself, have variant-specific vaccine formulations. Now, the success of any or all of these programs are going to be dependent on strain selection, some logical form of strain selection, known immunologic targets, available animal models, and some understanding of what gaps may exist within the phylogeny. And to solve those problems, I still think we probably need some basic virology campaigns on coronavirus virome discovery, receptor identification, which would help develop novel animal models to more distant antigenic strains that we currently don't have any way to query in terms of vaccine efficacy, importance for conserved epitope discovery, and for vaccine design strategies. Next slide. Now, using Pan-Sarbecovirus vaccine as a model system, I think we have clear evidence that this goal is likely very achievable. So, for example, our own group have been involved in 3 studies that have been published, all of which have shown use different basic strategies to provide not only protection in vivo against strains that are included in the vaccine cocktail, but also strains that are not included in the vaccine cocktail. And Kevin Sanders will be talking about strategies to focus immune response on conserved neutralizing epitopes a little bit later today. I'll mention briefly our multiplex chimeric Sarbecovirus spikes using mRNA-based vaccines. And later, David Wiesler, I think, will be talking about focusing immune responses on multiplex Sarbecovirus receptor binding domain based nanoparticle vaccines, all of which have provided in vivo protection. Next slide. An important consideration there, though, however, is that when you consider the breadth of the family, multiplexing strategies are going to be quite important, and that results in potential problems associated with immunodominance and balanced immunity across the vaccine platform. Now, as a model system, Sarbecovirus really does show what we need to develop for many of these other genus within the coronavirus family. We need high throughput neutralization assays. These are from my own laboratory against variants of concern, which are shown the 4 on the left, using a human BEI reference panel, a clade 1B related SARS strain from pangolin strain, which is actually more sensitive to neutralization. And then some clade 1 strains, including SHC014, a bat coronavirus, and SARS coronavirus, which are highly resistant to neutralization driven by either SARS-2 natural infection or the current vaccines that are being used against SARS-2. Importantly, we have robust animal model systems that fall into 2 platforms. The K18 model developed by Stan Perlman's group and others can be used not only against many of these variants of concern shown across the top left-hand panel, but also many of the zoonotic coronaviruses that use human ACE2 receptor cause, like SARS, like SHC014, WIV1, pangolin coronavirus, can all cause severe weight loss in the model. This is work done by David Martinez and Alex Schaefer. But we also have a large panel of mouse-adapted strains that cause highly significant disease in standard laboratory mice, providing models platforms for evaluation. Next slide. In collaboration with Sally Permer's group, we've also been looking at the effect of vaccine breadth in young primate infants vaccinated with either SARS-2 protein subunit-based vaccines in the blue dot on the top right, or mRNA-based vaccines. This is 8 weeks after infection. You can see against the standard Wuhan strain, D614G, you get robust neutralization titers, but as you move into more heterogeneous strains, like the zoonotic strains, shown here with SHC014 or Omicron, you see significant loss of neutralization. And Michael Mallory, who's doing this work, has followed these out through about 40 weeks, looking at the durability and breadth over time. This is on the bottom right-hand slide is some work done by David Martinez and Alex Schaefer in mice with SARS-2 based S2P mRNA vaccines. And you can see in the bottom left-hand corner that a Newton titer of about 1 to 10,000 is derived against the homologous strain. But as you move into these more distant clade 1A strains like SARS and WIV1, these neutralization titers drop 20 to 40-fold and up to 300 to 500-fold against SHC014. And importantly, SHC014 and Omicron are not the most antigenically distinct strains. There are other more distinct strains which we don't really have robust models for that are needed for Sarbecovirus. Next slide. Now, this is a strategy that we use, which is basically chimerizing spikes. It's based on the hypothesis, clear data from RNA recombinance that the spike is modular in design, and different domains like NTD, RBD, and S2 and S1 are interchangeable by recombination processes. In this case, you can design heterologous spikes that have the S2 domain from SARS-2, the NTD domain from a clade 2 strain, HKU3, and the RBD strain derived from SARS coronavirus. This could be multiplexed with spikes that have SHC014, RBD, providing 2 strategies. One, an immune strategy that focuses on the conserved regions in the RBD between these 2 strains, as well as enhancing type-specific responses against multiple strains. So any single spike could be considered as a 3-module unit, and with each multiplex, you can cover up to 9 different Sarbecovirus. Next slide, actually go 2 slides. One more slide, please. So when this strategy was used, go back one slide, please. So when this strategy is used, the SARS-2 mRNA vaccine protects against lethal SARS-2 challenge, that's the red dot. But these multiplexed chimeric vaccines also protect against SARS-2. When you move outside of that to a clade 1 strain, the mRNA-based SARS-2 vaccine does not protect against SARS-1 lethal challenge. However, these multiplex vaccines do, and you can see that, again, you have breakthrough infection, but not with the multiplex vaccines. Next slide. So some things to consider in spike vaccines is whether we want to use full length or RBD. I just mentioned two things about RBD that should be considered: the potential loss of quaternary epitopes, the potential loss of other neutralizing epitopes that exist elsewhere in the viral genome, and the potential for domain-specific targeting differences that occur across different clades within Sarbecoviruses. So the hypothesis now is that RBD is, in fact, the primary target, but that may not be true as we move across different coronaviruses within the family, and that needs to be researched. Multiplexing strategies need to be looked at for immunodominance, and a variety of heterologous prime boost strategies with different immunogens and different platforms, I think, is really critical. Finally, I think adjuvant considerations are important based on the work that we've done with Sally Permer. And also, we need to think carefully about immune durability, not only in the lower respiratory tract, but basic research into enhancing durability in the upper respiratory tract will be critical for control. Next slide, please. I think I'm about done. I just wanted to also mention T-cell immunity. This is some work that was done in Stan Perlman's lab to show that alpha virus-based vaccines with the nucleocapsid capsid protein of SARS or MERS can knock down virus replication. These are the two figures on the left. And also protect against lethal infection, although many animals die. So any sort of T-cell-based vaccine probably needs to be used in combination with a spike-based vaccine that elicits robust immune neutralizing titers. Next slide. And then some common obstacles. We need more high-risk strains, for example, not only in Sarbecoviruses, but other beta coronaviruses. For MERS, we only have MERS and closely related MERS variants. We need heterologous strains that actually replicate efficiently and animal models that exist that could be developed. The group 2D clade, among the beta coronaviruses, there are no strains that I know of that are replicating in cell culture or animal models that have been developed. The group 2A strains include the common contemporary human coronaviruses. There's limited reagents and animal models for them, but there are lots of animal models in 2A that can be used as surrogates and need to be considered. The alpha coronaviruses is probably worse off than the beta coronaviruses, besides NL63 and 229E, as the animal models are weak or non-existent. We need to identify high priority zoonotic strains, and that several animals strains certainly exist that could be used as surrogates and need to be considered. And finally, Delta coronavirus, porcine epidemic diarrhea virus, has been shown to infect infants, and so that needs to be included as well. And with that, I'll conclude with my final slide, just acknowledging people in my lab who've worked on this topic. Thank you.
Okay, well, thank you, Ralph. So in other words, there are multiple possible strategies invoking both antibodies and T-cell responses, which could then broaden immunogenicity. So that is, I think, an optimistic situation for a broader vaccine. So we will now move to our next speaker, who is Walid Abdulman Al-Jab from the King Fahd Medical City Institution in Saudi Arabia. Dr. Al-Jaber, can you take the floor, please?
Thank you so much for the invitation. Yes, my talk today will be about the clinical epidemiology of SARS-1, MERS, and COVID and other respiratory coronaviruses. So I would shed some light about the background overview and the emerging of respiratory coronaviruses that include MERS and other coronaviruses and symptoms and transmissions. So as you know, the emerging viral diseases and other coronaviruses is a major threat to global threat that can affect health. So there is a rapid mutation, adaptation to change in the environment, several new viral diseases and emerging and causing human illnesses. And also, as you know, with other all the viral diseases or viruses, they have the polymerase, that enzyme that help in viral replications. And also there are other factors in terms of biological behavior and ecological factors that contribute in emerging and re-emerging of viral infectious disease. So there are different way for the transmissions from the wild animals and domesticated animal with attended human diseases. So most of the viruses and coronaviruses come from zoonotic disease, or from they call it zoonotic infectious disease. Detecting new viruses nowadays, it's become easy with the technology that we have nowadays. So this is some about the history about the diseases and how these viruses come from. As you can see in this percent of viral, that most of them, they are from viruses. And they are, as you can see in this slide, most of them, they can be zoonotic and from wild animals. This slide show the viral or the coronaviruses and the year that they are infected, and the host or the animals that's been detected and where exactly in the country. So those highlighted those coronaviruses. So in a basic way, just to share what exactly or what's the main of these viruses, where they are come from. So you can see there are two main reasons for virus emergence. One from virus mutations that we call it genetic, and the other one call it changes in contact between human and infected vector or host, and then we call it behavior. So I will focus here about the second one, which about the behavior, and there's changes in temperature or the rain or whatever that have a new contact between host and human. The second part from the behavior is about the bushmeat and the live animal markets that allow contact between humans and infected animals that would otherwise not occur. For example, we have here Ebola and HIV and other, for example, monkeypox that allow new contact between virus and humans. This also with the animals or live animals markets, where we have SARS, MERS, and SARS-CoV-2. For example, about SARS-1, it was originated in animal market in China, that 2002, and the host was civet cats suspected, and the disease, as we know, it's contained in China, then suddenly become pandemic, and the cases or total cases about 8,500 and 900 deaths worldwide. Here is just more details about it. To move to this MERS, it is a novel positive sense single-stranded RNA of the genus beta coronavirus, and it is initiated or like called in novel coronaviruses in 2012 and been reported in Saudi Arabia. And the person was like fell in when he has like a contact with camels. It appears that some people became infected after contact with camels. And this in general about the symptoms, which are similar with other coronaviruses. that share with the cough, fever, and sore throat. This slide it shows which exactly confirmed contact on transmission. Those solid arrows, there is evidence of a transmission, whereas those dashed arrows, it's no enough evidence, or there is like no enough material, information about transmissions. Here about the primary, when it is there is no contact from with the humans cases, and the secondary transmission when it is between or from contact with a human MERS cases. Here about the environmental transmissions that the MERS has isolated from bed sheets and also IV fluid, suggests there is a potential for environmental transmissions. Regarding the viral shedding, we know RNA and the live virus found in upper respiratory tract and lower, and there is it is reported high RNA level in lower respiratory tract. Here, this is the most information about MERS. We have about 2,585 cases. 2022, there's no cases has been reported, and the death rate is about 37%. In terms of treatment, there is no specific antiviral recommended for MERS-CoV. In terms of vaccine, 10 years have passed since MERS discovered in 2012, and still there is no approved vaccine. There are some candidate of vaccine, but some of them still in phase 1 and phase 2. When we talk about SARS-CoV, as we know, it is reported in December 2019, and as we know, and I know some of my colleagues who will talk about this COVID more, and well, I think there's a talk about evolution of SARS, so I will leave a stage for him. But we know there's by March 2022, we have about 472 million confirmed cases that include about 6 million deaths. And here is about the phylogeny, about all the SARS-1 and MERS and SARS-CoV-2. And here's about the transmissions from coronaviruses from the bats to maybe either to animals or to the other in intermediate hosts then to the human. So in summary, there's a similarity of clinical features between SARS-CoV and other previous beta coronavirus infections have been noted. In cohort, most patients presented with fever, dry cough, and other symptoms. These features of SARS-CoV infection bear some resemblance to SARS-CoV and MERS-CoV-2. However, few patients with SARS-CoV-2 had prominent upper respiratory tract signs and symptoms of sore throat that include indicating that the largest cells might be located in either or in the lower airway. Also regarding the field and epidemiologic study lead to the initial genetic characterization of the virus and a description of the clinical and epidemiological feature of infected person. It is really important, and about the characterization of viral transmissions with different populations. Here's some our recent publications in COVID and other coronaviruses. Thank you so much for your attentions, and here our collaborations. Thank you so much.
Thank you, Dr. Al-Jaber. So in effect, we have a veterinary coronavirus that could be controlled by the same means that we are controlling other coronaviruses, and a veterinary vaccine or multiple veterinary vaccines could potentially solve the problem. So I look forward to the licensure and full use of those vaccines. Thank you. So now the next talk is on the evolution of SARS-2, and it will be given by Dr. Marian Koopmans from Erasmus because of the last minute inability of a speaker. So we appreciate Dr. Koopmans taking on this task with such short notice. Thank you very much. And can you start your talk?
Yes, thank you. If everything is all right, you can see my screen, right?
Yes.
Yes, okay, so if this seems a bit rushed, it is because it is, as I was asked to give this talk slightly 2 hours ago. So just some considerations from viral evolution studies that I think are relevant given the topic of today, which of course is Pan-Sarbeco vaccines. So that this is looking at the, I would say, one of the positive aspects of this pandemic, which is just the amount of genomic sequencing that really has allowed us to put a magnifying glass on one of the best studied emerging disease outbreaks of our day and age. And this has really generated new insights in evolution of an emerging virus and has been used for many questions, including how do we, can we assess transmissibility, antigenicity, virulence, and treatability, which of course are somewhat linked traits. And what we have seen with this pandemic is variant emergence caused by different and complex mechanisms. Of course, we are looking at coronaviruses, which were thought to be not so rapidly evolving because of the presence of a proofreading mechanism, and therefore with lower frequency mutations and than for instance for HIV and influenza. But nonetheless, over the course of this pandemic, of course, we've all learned that there is still active evolution ongoing. So this has been witnessed very much with the emergence of variants of interest and variants of concern, two now definitions used by the WHO. Variants of interest are defined as viruses that pop up because of increased circulation in more than one region or unusual outbreaks. And with some mutations that may raise concern because of the location where they are, and then variants of concern if some of those mutations are associated with key traits like the ones I just mentioned. And we have seen that process, which has come as a bit of a surprise, I think, to the global community, here with the example of the birth of the alpha virus variant, and here with Omicron, which after the sequel of viruses until then, including Delta, really seem to have branched off quite early on and now is circulating as a highly diverged virus. So the question still is very much where do these variants come from? Are they new introductions? Do they evolve within immunocompromised host or do they maybe come from animal passage? And those are, I think, still outstanding and competing hypotheses, which may all be at play for all we know currently. The first, of course, being that there may be pockets where these viruses are circulating among humans, but where there is insufficient or far less coverage of the genomic surveillance initiatives because that is the case in quite a wide region of the world, and where we may have communities where these viruses have evolved, but that are not so connected globally until viruses then are introduced into more densely populated regions. One hypothesis that still really needs to be looked at. A second one is, of course, the animal passage hypothesis, and examples there are from the observations that we've seen viruses introduced from humans back into different types of animal groups. Here into mink in blue, with several separate introductions into mink farms in this instance in our country, in the Netherlands, and then human to mink to human to mink to human transmissions that were associated with changing in the receptor binding domain, and that were only stopped by complete elimination of the farming sector in this case. And we also have the somewhat worrisome situation of repeated introduction of SARS-CoV-2 into deer populations, at least I think 5 or 6 separate documented introductions again, with recent documentation of a highly diverged virus circulating among deer and associated with spill back into humans. So the animal passage as a potential source of new variants certainly cannot be ignored. And the third hypothesis is somehow evolution of these viruses in immunocompromised patients. There are several anecdotes out there, and here's an example just from very recent, where an alpha virus variant popped up in surveillance in our country, where alpha viruses have not been detected since October 2021. But this virus had continued to circulate in a patient with chronic leukemia, and in retrospect, that person had been infected with almost for now over 300 days, and the virus has picked up several additional mutations, again, pointing at a risk for further evolution and reintroduction. Now, of course, the real challenge that we had and still have, I think, is that we are looking at very fast expanding range of virus after introduction of the massive global dispersal with multiple seeding events, which by themselves, just by the nature of that, can lead to genetic lineages without there being any difference between those viruses, but then also the kicking in of immune selection pressure further advanced by the introduction of vaccines, when we started to see selection, of course, for immune escape variants. So with that increasing immune pressure, of course, also the risk of immune escape increases. Now, that's what we've been seeing now with most clearly with the emergence of the Omicron variants. And it's very easy to see just how diverged these viruses are by looking at this alignment of the spike protein, where you see in color all the different mutations, including mutations in important in the receptor binding domain. What we also now see is that if you use the types of methods that had been developed to map or characterize antigenicity influenza through antigenic cartography, and you apply that for SARS-CoV-2 here through hamster immunization studies, we are now, I think there is increasing agreement that we may be looking at separate serotypes by now, where the majority of viruses pre-Omicron sit here, and Omicron BA1 and BA2 are quite distantly related, leading to the discussion what that means now for updating of vaccines, or maybe moving forward with two-component vaccines. Important in all these studies and all the studies that are done to then characterize viruses and responses is that we may call something Omicron or Delta, but there's actually a wide array and diversity of sublineages, which also are characterized by a whole range of mutations outside the spike protein gene, which I think we still need to learn about, just the relevance of that. So deriving conclusions from experiment with specific viruses really needs to be done with caution at this stage. So with that, I've just summarized that, of course, this virus is evolving, but the direction of evolution, we are really standing in very early stage knowledge about this. Will the next variant be derived from Omicron or back to the root from a previous virus? I don't think we can be sure there. Sources of emerging variants really need to be resolved, and the different hypotheses, in my view, need to be included in enhanced surveillance. And we are at the start of discussions on vaccine updates. And I do think that the thinking about broader vaccines, pan-coronavirus vaccine, can also benefit the current SARS-CoV-2 response.
Well, thank you very much, Dr. Koopman, for that presentation on short notice. You know, personally, I think I'm in favor of the immunosuppressed individual hypothesis, if only because that has happened with rubella, attenuated rubella virus that is reverted in people with certain lesions. Well, we'll go into the details. So the next speaker is a long time. Next speaker is William Karish from Echo Health, and he will speak on geographic and host distribution of coronaviruses.
Great, thank you, Professor Salk, and greetings, everyone. If you could we could just jump right to the next slide. So as to kind of give a review, as you just said, with the geographic and host distribution. And a month ago, Professor Kristen Drosten presented this slide, and I was very quick, and I did a screen capture, and I stole his slide, and I was going to start my talk with this, because I think it's important to think about, and this is really from Felix Dressler originally, Professor Dressler, on this expansion of knowledge that we've seen in learning about coronaviruses across different hosts and geographic regions. And had you asked me to kind of give this presentation 10 or 15 years ago, it would have been very simple. But what we've seen 10 years after the original SARS is this great expansion of knowledge about coronaviruses and what's circulating out there. So I'm going to kind of focus in, and Professor Barrick was kind of talking primarily about the alpha and beta coronaviruses. If you go to the next slide. This is a study just at a global level, mostly around the global tropics, looking at coronaviruses in animals in general, though this has some human viruses pulled out of GenBank in there. The yellow ones are non-bats. And then the others are all from bat families, and this is kind of covers probably 100 or so coronaviruses found, in alpha and beta coronaviruses found. And if you go to the next slide, great, this is kind of some of the mapping of the geographic distribution. You can kind of ignore the hotspots map. The red and the green have to do with the host distribution. But those dots are showing individual alpha in the upper left, the alpha, and those are alpha coronaviruses and beta coronaviruses. So you kind of see this kind of global distribution that was found, and this work is over the last 10 years, just uncovering about 100 or so previously uncharacterized coronaviruses. Go to the next slide, please, to get us back in thinking of that. So within the beta coronaviruses, I think was already presented earlier, you have these different lineages. They now kind of, I'll talk about them in these, in their kind of subfamily names or group names. But you have this lineage B up on the left, which is the Sarbecoviruses. But we also have these others. And if you can go to the next slide there, we kind of really expanded on that knowledge. So some work that's being really done now. This is courtesy of Dr. Cecilia Sanchez, and it's coming out in a paper, which I'll reference soon, but it's available, kind of looking at just within the bats, the different subgenera for the beta coronaviruses. So this is Sarbecoviruses. You see this kind of group of great distribution of different bat species. These are mostly fruit bats. In this group, you can see Asia and Africa, and into Australasia, Australia. Next slide, please. The Sarbecoviruses, another group. This is a lot of these were being found in large fruit bats, the pteropods or flying foxes, but those are all individual species. And then, so this is really just compiling the work, and all the references are available for each one of these lines about who was able to identify that virus or pick that up. Once again, you see kind of an Asia and Africa distribution for this group. If you go to the next slide. The Sarbecoviruses, so these are particularly in insectivorous, small insectivorous bats, and you get this little broader geographic range of Asia, Africa, Eurasia, Europe, even Latin America, North America, into the Americas. And then the next slide, please. And then down into the ones we're kind of really focused in on more today because of SARS-CoV-2. This beta coronavirus, the Sarbecoviruses. And once again, we still get another kind of broad range of species. This group, this subgenera has been identified in there, particularly in insectivorous bats. Not exclusively, but mostly in insectivorous bats. And once again, Africa, Asia, Europe, Eurasia, there. I would like to note, I mean, as we've kind of discussed on these other things, especially with some of the insectivorous bats, and Felix Drexler brought this up many years ago. When you think about these viruses, and you've been in our previous presentations about the variation within the one SARS-CoV-2 virus that we're all talking about, and this genetic drift and the changes in the different types of those viruses. We think of bats living in basically their own urban environment. Some bat colonies are 5, 10, 15 million bats living together in a colony. So that opportunity for these viruses to change and evolve really quickly within even one species of bat, and we have over 1,200 species just of bats, and we have over 2,000 species, 40% of all mammals are rodents. So we're just really looking at the tip of the iceberg on the potential coronaviruses in bats and in rodents, and then in other animals. Next slide, please. This analysis was also out as a preprint. We can use it, and those tables I was just showing you kind of underpinned it. But this is kind of a distribution of Sarbecoviruses for the host. So one thing is the redder, more purple area is where there is more diversity, the number of species of host. And then that's kind of plotted against the feasibility, the challenge of being able to sample those animals. So we're kind of talking earlier about challenges in vaccine development. There's also some challenges and opportunities for this kind of viral discovery to fully understand more about the diversity of coronaviruses. Some places it's easier to do, some places it's more difficult to do, and that has to do with accessibility to where these animals, wild animals, live, or the current lab capacity there, or training needs. So I would also say there's a great opportunity to expand our knowledge by thinking about where these bats, where the viruses probably can be found and where we could do more to find them. So next slide, please. This is just, I'm going to show you two quick ones for the Sarbecoviruses. This is a projection with climate change out the next 50 years. This is species richness. It's not the viral richness. It's this host species richness once again. So with climate change, we're predicting for Sarbecovirus bat host, you'll see the species richness going down in Europe. And next slide. But we're not seeing that kind of potential change over the next 50 or 60 years for Asia. So I think we'll be dealing with this potential for viral spillover, evolution going on for the foreseeable future. Once again, reinforcing this need to think more clearly about a pan-coronavirus vaccine, if that's a useful thought for you. And next slide. Particularly for SARS-CoV-2, now we've got a range of host species just for this one particular virus. So this has come out, so I didn't credit, this is Professor Jürgen Richt. I kind of shared this and put this together with colleagues. So we see a broad range of animals, and Professor Koopmans just mentioned this too about the SARS-CoV-2 virus getting into a variety of animals. This is a mix of human to animal infections, but also experimental infection and looking at host susceptibilities. Pretty broad. It starts to remind me of rabies. When the rabies virus we know affects over 120 species of mammals, including humans. Next slide, please. Just once again, we're not getting into it, but even for this particular virus, we're kind of seeing this animal crossover and some of the links there. And then finally, I'll close with the last slide to try and get us back close on time. This is official reporting for SARS-CoV-2 in animals, official government reporting to OIE. We see, and this is over a 2-year period, it's still the ability for countries to report, or the willingness. I think a lot of it's the ability to actually do the testing and to report on the presence of SARS-CoV-2 in animals is still quite limited to countries that have those capabilities. So once again, I think there's a need, if we're going to understand even SARS-CoV-2 in animals or host distribution, but all of these other coronaviruses, we're going to have to think really hard about engaging the capabilities of more countries to be able to contribute. On the next slide, and I'll close with that. Thank you very much.
Well, thank you, Dr. Karish, and thank you for showing us the size of the problem and the necessity to realize that there will be more Sarbecoviruses getting into humans. All right, well, now we will hear from a longtime worker in the field of coronaviruses, Stanley Perlman from the University of Iowa.
Okay, thank you, Stanley. So today what I'm going to do is talk about clinical characteristics of coronavirus infections. And can I have the next slide, please?
Apologies, my screen just froze. Okay, sorry.
Okay, can you make it? Thank you. Thank you. So what I'm going to do is compare mostly the SARS coronavirus, MERS coronavirus, and SARS-CoV-2 infections, and point out some similarities and also some differences. And I think this is important as we go forward in developing vaccines, because we want to understand exactly what we're trying to prevent. And also there are subtleties between the different infections, and we need to think about that as we develop vaccines as well. So what is common to all these infections, of course, is severe pulmonary disease that occurs in a fraction of people. The incubation periods are pretty consistent of about 5 to 7 days, though it's been noted that, of course, with the new SARS-CoV-2 variants of concern, the incubation period is a little shorter. And there's also other differences. So patients who have MERS ended up in the ICU much more quickly than those who had COVID-19 or SARS. And from some studies, I think human challenge studies, and I'm not sure they're published yet, they show that virus after exposure can be detected in nasopharynx within just 2 days. So this virus goes into the nasopharynx, and SARS-CoV-2 replicates very quickly. Of course, this information is not available for SARS and MERS. There are specific and non-specific symptoms associated with all these diseases, including cough, fever, myalgias, shortness of breath. And many people infected with all three entities have mild disease. But here there are some differences, because for SARS, it was said that there were no patients who were asymptomatic. Well, that's clearly not true for COVID-19. For MERS, it's a little trickier because there's not many when you do epidemiology studies, but there's clearly people who are infected and then clear the infection quickly. In all of these cases, the people who are at greatest risk are those who are over 75 and those with comorbidities, diabetes, obesity, lung disease, heart disease. All these viruses tend to be opportunistic and attack people who have less ability to defend themselves. Diarrhea and detection of viral RNA in the feces is common in all three diseases, and renal dysfunction has been reported in all, though it may be greatest in MERS. This is something that's a little unclear. In the beginning of the MERS epidemic, it was clear that there was a lot of renal disease, less clear now that that's a very, very common feature. Can I have the next slide, please? And this is just to point out, this is a slide from a year ago, and it's one of my favorite slides, because what it points out is that taking the standard group of 18 to 29-year-old individuals, those who are 85 years and older have a 635-fold higher incidence of death. So this is hugely different, whereas children really are much more likely to not get serious infection. This, of course, has made it a controversial issue, at least in the United States, about whether these children should be vaccinated or not. Controversial to some people. Could I have the next slide, please? There are several laboratory features that are also common in these three entities. Lymphopenia, or a low number of lymphocytes in the blood, is found in all three diseases, but it's more common in SARS and COVID-19. The information is a little unclear in SARS because all patients were treated with steroids, or basically all patients. And in MERS, the same thing. Many patients were treated with steroids. Neutrophilia occurs in COVID-19. It was not noted in SARS or MERS. And the neutrophilia may not be found in all patients, and it may have more to do with the presence of more activated immature neutrophils in the blood and in the lungs than in actual neutrophil increases being present in all patients. Chest X-rays and CT findings are consistent with a viral pneumonia, but they're really not diagnostic. They were useful early in the pandemic in China because most patients who were coming in with viral pneumonia had COVID-19, so therefore it wasn't unreasonable to assume that somebody coming in with a pattern that looked like viral pneumonia actually had SARS-CoV-2 infection. And then the other thing we know is that myeloid cells are infected, but abortively, but this may contribute to the pro-inflammatory response that's so common in COVID-19. Can I have the next slide, please? And so how do the features differ? Well, the most important one I don't even have on the slide here, which is that the upper respiratory tract is infected profusely in COVID-19, not so in SARS and MERS. And this, of course, is why we have a pandemic, because the virus transmits much more readily, particularly in people before they're symptomatic, the pre-symptomatic people, and perhaps even asymptomatic people. SARS and MERS only transmitted after pneumonia was recognized, so it's much easier to quarantine those people and prevent them from infecting others. The mortality is quite different. In SARS, the mortality is 10%, MERS 35%, COVID-19 1 to 2%. But one thing that I think is interesting is that if 10 or 20% of COVID-19 patients develop severe disease, and 1 to 2% overall mortality then is equivalent to the 10% mortality that we see in SARS if we count only the people with severe disease. So again, a similarity between these infections. Could I have the next slide, please? And other features that are shared by both SARS and COVID-19 are that in both diseases, multiple organs are affected. This may be true in MERS as well, but we know less because there's only been 2 autopsies of MERS patients. So we have less information about what organs are infected. We know that from at least some studies, SARS-CoV-2 was found in many organs, but this was not really confirmed in other studies. And it's an issue because in SARS-CoV-2 and COVID-19, virus is not commonly found outside of the respiratory tract. There's scattered reports that it is, but I don't think it's convincing. So we don't understand why there's multi-organ disease when there's so little virus found in these other organs. So some of the things that we see in patients who died from COVID-19, we see effects on pneumocytes, we see embolic phenomena, we see brain infection, endothelialitis, and this may be the key thing. There may be effects on the endothelial cells that contribute to all these other organ dysfunctions. This is something we need to know about because this may impact the way we think about vaccines. And we see other entities such as megakaryocyte or platelet recruitment and hemophagocytosis. Could I have the next slide, please? And then, of course, there are some things that seem to be unique to COVID-19, not completely clear because we know so little really about SARS, but neurological symptoms that occur soon after the acute infection, which include brain fog, memory loss, confusion, anosmia, ageusia, and stroke, heart problems, including myocarditis, rashes that often take a long time to resolve. And when we look at people who died from COVID-19, we see many of the same phenomena. We see thromboemboli, we see brain infarction, we see endothelial cell damage, kidney damage, splenic damage, and hemophagocytosis, megakaryocyte recruitment. So many of the same things that I mentioned already are also found on autopsy. And again, we don't understand the pathogenesis because virus is, for the most part, only found in the lungs. Can I have the next slide, please? And then finally, really the last part of thinking about clinical disease, because this is not just an acute clinical disease that goes away, is we have to think about long COVID. And this may be the most important thing in the long term that we have to think about in how vaccines will ultimately help us. So long COVID has been slowly recognized over the last 2 years. It may have some similarities with encephalitis lethargica, which followed the 1918 flu, but encephalitis lethargica is even being revisited, and there's some thoughts that it really wasn't directly a result of the influenza infection. So it's not totally clear to me how it occurred. The long COVID symptoms include some of the things we've already mentioned: cognitive losses, mood changes, prolonged anosmia, tinnitus, and headaches. That was another thing I should have mentioned that's really unique about COVID-19 is the loss of sense of smell and taste. That's not found in SARS and in MERS, though I have to say in our mouse models, we do see anosmia in all 3 virus infections. Pulmonary sequelae are also noted, the shortness of breath and coughs, and there's some cardiac problems, including POTS, which is pretty common, posterior orthostatic tachycardia syndrome. And then I just recently read about increased risks of developing diabetes in those who recover from COVID-19. And the disease has many similarities with the chronic fatigue syndrome. So we think the vaccination will protect against long COVID. But one thing is, since we don't understand the involvement of organs other than the lungs, and we don't understand long COVID, we do have to keep our eyes open about whether vaccination itself could mimic some of these effects, because if it's just antigen, could that occur in vaccinated people? Something to think about. So far, not much evidence that this is a problem. So can I have the last slide, please? And the biggest concern is that long COVID will not resolve in some patients, and will there be increased incidences of neurodegenerative disease, particularly in people who have anosmia or ageusia? And as I said before, vaccination likely will protect against long COVID. However, if viral antigen is a contributory factor, the effects may be nuanced. Okay, with that, I'll stop.
Well, thank you very much. That was a brilliant coverage of all of the clinical symptoms of the COVID disease. And now we move to different strategies for vaccine development. And we have a number of speakers, each of whom has 10 minutes. Please keep to your time. We'll start with Linfa Wang from Duke in Singapore.
Thank you, Stanley. So I'm going to talk about Pan-Sarbecovirus booster vaccination. So it's very important. We're talking about Pan-Sarbeco. Florian and Dr. Kramer will talk about differentiation between truly Pan-Sarbeco versus so-called variant proof. So I want to emphasize our strategy is truly Pan-Sarbeco, and also we're focused on a booster vaccination rather than a primary. Next slide, please. So can we ever be able to succeed in making a pan-something vaccine? That has been a dream. So next, you can keep click here. So Pan-Sarbeco is the dream, pan-flu is the dream, right? And pan-dengue in my region in Southeast Asia. Next one, we want to, next please. Pan-coronavirus, next. Pan-Sarbecovirus is the topic of today. But before that, I did succeed in make a pan-something virus. Next. It's the pan-Hannipaviruses. Next slide, please. So Hannipavirus is another class of deadly bat-borne virus, from bat to horses to pig, and then human-to-human transmission. So we first discovered in Australia the Hendra virus, led to the Nipah in Malaysia, Nipah, Bangladesh, and so on. So we developed a vaccine. Click, please. Next is a vaccine that targets horses. It's a soluble equivalent of the spike protein we call the G protein, the envelope protein, and it's protein subunit vaccine. Really, it's a One Health approach. We developed that vaccine next to prevent the transmission to humans. Next slide. It was very successful and became the first licensed vaccine. Have a steroid immunity. Basically, if you vaccine the horse, they will never get any replication, and let alone shedding and the transmission. Okay, so it got approved for horse, and it was the first ever approved vaccine against any biosafety level 4 agent. Next. To our pleasant surprise is that the same subunit vaccine without any modification, when we immunized in experimental animals, it was 100% protective against the Nipah virus, which is the same sort of two different viruses in the same genus. And next. So this is the formula now. CEPI is funding several Nipah vaccines. One of them is actually based on the soluble G protein from the Hendra virus. So I just want to demonstrate that it is possible to make a pan-genus vaccine if we have the right strategy and also have to be lucky with the right virus. Next. Now, if you compare the amino acid sequence identity of these two key antigens, right? For Hannipavirus, it's the G protein, and for SARS and MERS, the coronavirus is the spike protein. So both are receptor binding envelope proteins. You look at the sequence of homology, it's almost identical, 78% identical between Hendra and Nipah, and 76% between SARS-1 and SARS-2. And very early on, when I came back from Wuhan in January 2020, and immediately in Singapore, we recall back the SARS survivors, and we demonstrate the SARS survivors still have neutral antibody against SARS-1 17 years later, but totally useless in terms of neutralizing SARS-CoV-2. Next. Now, we are really sort of puzzled, right? With Omicron, it's so potent in escaping the neutral antibody induced against vaccine or infection against the ancestral virus. And you look at the sequence identity, it's 97.7. Next slide, please. So the conclusion is clear. The coronavirus protect immunity, especially those mediated by neutral antibody, is very much more virus or strain specific than the other family I worked, the paramyxoviruses. Next. So now, in order to induce a true Pan-Sarbecovirus vaccines, I think we only have really two realistic strategies. One is a mixing of the virus-specific neutral antibody. So we have a cocktail, different spike to be different IBD against different virus. You can mix Omicron, ancestral SARS-1, and hopefully you basically have a cocktail of the antibodies. This neutral antibody is still virus-specific, but now you have a mix so that the synergy, you put them together, you get this Pan-Sarbeco. Next. What we are trying to do is the induction of cross neutralizing antibodies by sequential and cross-clade boosting. Next. So, the immunodominant epitope from SARS-1, we put this in the circle, and then COVID is a triangle I presented in the last WHO meeting. So in order to induce this cross-neutralizing, we hypothesize, you need to do cross-clade. Next slide. And very luckily that we proved that, in our publication now more than half a year ago. And next, out of 5 panels of serum we used, there's only 1 panel truly developed this Pan-Sarbeco neutralizing antibody. And the assay we used is the surrogate virus neutralization assay, which allow you simultaneously in the same tube determine neutral antibody against 10 different Sarbecovirus in this study. And we now have gone up to 21 different Sarbecoviruses. Next slide, please. So very encouraged from this first in-human sort of data, and we went ahead and designed a Pan-Sarbecovirus booster vaccine strategy. And the booster vaccine candidate was designed. It's an artificial sequence, does not exist in nature. It basically is a consensus sequence, covers all the known clade 1 SARS coronavirus, and mainly from bats. Okay, and priming mice and with 2 doses of the mRNA vaccine. We did the priming using 2 different mRNA, the Pfizer, Moderna, and also the Sinovac inactive virus. But here I'm going to focus on the BNT vaccine. And the cross-clade boosting was achieved with 2 formulations. Both are protein subunit vaccine. The first one is the in-house trimeric S, and we use 25 micrograms. I mean, it's almost overdose, because we don't know if it's going to work. And we use the experimental adjuvant we just ordered from Sigma. Later, last few weeks, we just completed a second, boosting study. This time using a GMP cell bank. So it's already GMP sort of ready to produce it. And it's a trimeric, but it's 1 microgram this time. Instead of using the experimental adjuvant, we use approved human adjuvant with a pharma collaboration. All groups produced Pan-Sarbecovirus neutral antibodies. And then we now assay these neutral antibodies using our latest multiplex. The New England Journal paper, we use a 10-plex. And this study, I'm going to present it use 16 plex. And then just last week, we have increased to 21 plex. Next. So this is just to give you really a very kind of a brief summary. PPP means that you have 2 Pfizer vaccines and followed with the boost, also a Pfizer mRNA, so PPP. PPC is the priming with 2 mRNA and then boosting with the consensus vaccine, use 1 microgram. PPC 25 is the first experiment we did, use our in-house spike protein and use the experimental adjuvant. PPC 1 macro, no adjuvant, is just 1 microgram without adjuvant, and PBS is a negative control. And we have day 7 for most of them, but for 1 group we don't have. And then for day 14, we PPC 25 we don't have. So don't worry about the specific virus. I will come in the next slide. I just want you to focus on the pattern. Sorry, just stay on the slide, the previous slide. The pattern is clear. If the curves are broadened like the PPP, it means that it's not a very Pan-Sarbeco neutral antibody. And when you get tight, like the PPC at day 14, as you can see, it gets to really tight, close together. So that gives the breadth. If you have a broad Pan-Sarbeco vaccine, you should neutralize the virus all very efficiently. And then in terms of the absolutely tighter, then the more shift to the right, then have a high titer. Next slide, please. So now we're basically still PPP, PPC on top. It's the same kind of labeling. And now we're presenting the data for each individual virus from SARS-CoV-2 on top to the SARS-CoV-1 at the bottom. It's a 16 plex, 11 is clade 2 virus, and the 5 is clade 1 virus. So just focus on the first 2 columns, PPP versus the PPC 1 microgram, because the time is very limited. So now you can see that at the day 14 bleeding, the last column of each group, you can see for SARS-CoV-2, the red is higher than the next column, so the kind of a greenish color. And it's also true for the second one is alpha. But the third one is delta, and now you see the PPC is catching up. And then for the remaining of the viruses, PPC is all better than PPP. So the homologous boosting with 3 mRNA vaccine only give you high antibodies against the very homologous virus itself and alpha. For everything else, the cross boosting with our consensus protein subunit is better. Next. So these are the numbers, and I have to, really emphasize this is a one-off, n equal to 5 from each group, and we have not had the time to repeat this yet. So don't take it for, sort of conclusive, but the trend is definitely there. So you look at the absolute numbers of the neutralization titer at the 50 with our surrogate versus neutralization, and then the PPP and the PPC. The third column is the ratio, the PPP versus the PPC, and then you look at the SARS-CoV-2, it's above 6-fold. So the absolute neutralization titer for the original virus, 3 doses of mRNA is better, but the PPC is better for every other virus. Okay, so I just list a few, but the other 14 virus. The other way to look at it is the deviation from the original virus, how much reduction of the neutralization. So for SARS, the PPP, as you can see, that it at least 70-fold for Delta and it goes to 1,000-fold for Omicron. And then for PPC, Omicron is still the worst, but it's not that much different from the original homologous boosting. Next. So to summarize, what we basically have demonstrated, cross-clade boosting produced broad neutralizing antibody for all known Sarbecovirus. Every virus, we have unpublished virus as well, from bats. As soon as we know the virus, the RBD can bind ACE2, we can put into our assay within 2 weeks. The PPC 1.25 boosting is better than the PPP for all 21 viruses except the ancestor and the VOC. So I presented 16 viruses, but now we have 21 viruses. So only the ancestor of alpha, the PPP is better. The C1.25, the consensus vaccine candidate boosting, has significantly increased the breadth, but there's still room for optimization to increase the absolute neutralizing antibody titer, because we only use this 1 microgram with the approved human adjuvant, and this is only one experiment. Next. So the future kind of ongoing projects, first of all, is that if mRNA can give you high neutralizing antibody, then what happens if we use the C1.25 mRNA, use the Pfizer platform or Moderna platform? So we're working on that. And then other protein delivery system. For this experiment, it's a soluble full-length spike. We know the nanoparticles of VLPs maybe can increase that. And the dose optimization, the comparison of the different adjuvants, certainly there's a room, right? Because our 25 microgram in-house soluble soluble spike with an experimental vaccine already better than the 1 microgram with approved human vaccine. So the dose and the vaccine adjuvant combination can certainly further improve that. Thank you.
Okay, thank you, Dr. Wang. So the next speaker is Kevin Saunders from Duke in North Carolina.
Sandy, apologies for interjecting. Just a request for the speakers, if you can get to 10 minutes, but better if you get to 8 minutes, it will be better so that we can have time for some discussion. Apologies for interjecting.
Okay,
because we have more speakers than anticipated. Apologies. So please, all the speakers, only 8 minutes if possible.
Okay. And so I'll start right in. So I want to talk today about strategies for a pan-coronavirus vaccine that we're working on here at the Duke Human Vaccine Institute as part of a project grant that's funded by NIAID. And so we're approaching this from multiple angles where we believe that we're operating on the hypothesis that activation of multiple arms of the immune system will lead to the broadest protective immunity. We think that we're going to have to combine protective T-cell responses, protective non-neutralizing antibody responses, as well as neutralizing antibodies to conserve sites. So today I'm going to focus on neutralizing antibodies to conserve sites just for the sake of time. We've shown previously that we can multimerize antigens from different viruses onto the surface of protein scaffolds and augment the human immunity that's raised against that particular antigen. So we frame shifted this approach to SARS-CoV-2 at the outset of the pandemic. As diagrammed on this slide, what we did is we took the ectodomain from SARS-CoV-2, we added a 6 amino acid sortase A tag to that protein. And this allowed us to then conjugate this protein to the surface of a protein scaffold. In this case, it's a ferritin nanoparticle. This forms virus-like nanoparticles, as you can see diagrammed here on the right-hand side. And by negative stain electron microscopy, we can visualize those and show that we indeed form these types of molecules. As we were making these types of molecules, there was a lot of work done on neutralizing antibodies that had some demonstration of breadth. As diagrammed on this slide are structures of different neutralizing antibodies and their epitopes bound to the RBD. As you can see diagrammed here, the RBD is shown in black, with these different colors representing the FAB or antigen binding fragment of different antibodies. I've labeled here in red the antibodies that are cross-neutralizing antibodies, meaning that they can neutralize viruses other than SARS-CoV-2. The point of this slide is to show you that there's multiple epitopes on the receptor binding domain that can be targeted by broadly neutralizing antibodies. From that observation, we decided to also make nanoparticles that can display just the receptor binding domain. So we use the exact same technique where we can take just the receptor binding domain of SARS-CoV-2 or other coronaviruses and then decorate the surface of a protein nanoparticle in order to generate virus-like particles as diagrammed here on the right-hand side of the slide. As we did this, we then wanted to move in vivo and show whether or not these types of vaccines could protect against SARS-CoV-2 challenge. And so we made a couple different varieties of these immunogens. We made one version of the receptor binding domain nanoparticle that had the Washington 1 strain of the receptor binding domain. We also made one where we had the B1351 strain, or beta variant. We did the same thing for the ectodomain, where we had Washington 1 strain as well as beta strain. We vaccinated mice twice, as demonstrated here on this slide.
Dr.
And
Sanders,
this allowed us to then,
apologize. There is a request from the interpreters. If you can speak slowly, will be better.
Okay,
thank you.
I'll try. I thought I had 10 minutes, but if we only have 8, I'm gonna have to move a little faster. Okay, so what we did is we then challenged these animals with the mouse adapted strain of the beta variant, as indicated here with this red arrow. And so these studies were performed in partnership with Ralph Barrick's team and David Martinez. As shown here on this slide, just for the sake of time, we looked at initial weight loss as a measure of severe disease. As you can see, whether we used a receptor binding domain nanoparticle, diagrammed here in the red or the orange color, we can use one that's matched to the Wuhan strain, or we can use one that's matched to the beta variant. Regardless of which strain of the RBD we used, we provided protection against weight loss in these aged biopsy mice. I should point out that this is a lethal model, and so by day 3 and day 4, mice begin to pass away from infection. We also saw very similar results if we use the ectodomain. And so this demonstrated that we could use either receptor binding domains or ectodomains and generate protective immunity. These two platforms become great tools that we can now use for trying to broaden the antibody response. So for the antibody response, we are looking at 4 different ways of being able to broaden that response. So the first are mosaic nanoparticles, and this is something that's been talked about before by others, where you can display multiple coronavirus antigens on the surface of a nanoparticle. There's a second approach where we can also use epitope-focused immunogens, where we try to engineer the protein of interest such that it only presents cross-reactive epitopes and it has a dampened response against the non-cross-reactive epitopes. Third, we can also use the chimeric envelope design. And so this is work we've done in partnership with Ralph Barrick, and he mentioned that in his talk this morning. And then lastly, we're looking at different ways of priming and boosting, as you heard in the previous talk from Lin-Fa Wang, that if you can put together the right antigens as a prime and then different antigens as a boost, you may have an ability to actually broaden the antibody response. So first, I'll talk about mosaic nanoparticle immunogens. So just as an example of the way that we're using this technology, we've diagrammed here on this slide the concept of using a single receptor binding domain, diagrammed here as blue and red, that comes from SARS-CoV-2. To try to broaden the response beyond just SARS-CoV-2 or Sarbecoviruses, we can then move to a platform where we actually decorate the surface of the nanoparticle with multiple different receptor binding domains. In this case, we use the receptor binding domains from MERS, a bat coronavirus HHC014, and then lastly, SARS-CoV-2. We again did an in vivo challenge study where we immunized aged biopsy mice twice, and then we perform a challenge with SARS-CoV-1. And so this particular virus is not included in the receptor binding domains that's on the surface of our nanoparticles. As you would expect, when we immunize with either of these two nanoparticles, well, where the SARS-CoV-2 only nanoparticle is shown in blue, the trivalent the mosaic nanoparticle is shown in red, and then our adjuvant only controls are shown here in gray. We found that we could generate new IgG binding antibody in the serum for both SARS-CoV-2 antigen as well as SARS-CoV-1 antigen in both of these groups. However, only the mosaic nanoparticle immunized group shown here in red generated binding IgG against the MERS coronavirus ectodomain. We performed this heterologous challenge, and the data for that is shown here on the right-hand side, where we challenged the mice with SARS-CoV-1. As you can see in both groups, consistent with the generation of SARS-CoV-1 binding IgG, we saw protection against weight loss, whereas in our animals that were immunized with only the adjuvant or unimmunized animals developed weight loss in this particular model. So we can, again, provide protection against a virus that's not included in the vaccine is the point. We've also looked at ways of putting these together to broaden the response by boosting with different, priming and boosting with different immunogens. For the sake of time, I'll just talk about this concept where we are investigating both using SARS-CoV-1 and SARS-CoV-2 prime and boost as a way to broaden the response, as well as a way to focus in on the cross-neutralizing epitopes that exist on both receptor binding domains. To even move us another step further, we're also using this mosaic nanoparticle as a way of doing priming and boosting so that we can further the exposure of different receptor binding domains, but also again focus in on conserved neutralizing epitopes that would be present among the different viruses. In doing these types of experiments, we're hoping to learn the rules for which spikes work best as primes versus boosts as a way of understanding the amount of sequence diversity that can be represented on different nanoparticles. Third, we're looking at epitope focusing. And so what we've done is we've taken a couple of our different broadly neutralizing antibodies. Shown here on this slide is DH1284, also DH1047. These are two broad neutralizing antibodies, as you can see here, listed with their IC50s in microgram per mil. They're both broadly neutralizing antibodies. The main difference here is that 1047 is not active against BA1, whereas 1284 is. However, 1047 does have activity against multiple other coronaviruses, including SARS-CoV-2, CoV-1, WIV1, and SHC014. And so we use these as a template for designing different receptor binding domain molecules. And we do this by first solving the structure of those antibodies bound to the coronavirus ectodomain. Then once we know the footprint of those antibodies, it's diagrammed here with the receptor binding domain in black, and then their binding sites shown here in green for 1284 or 1047. We can then generate engineered proteins that then focus in on this particular site as opposed to the other sites on the receptor binding domain. We've also taken a similar strategy for the stem helix peptide based on work from David Wiesler's group that have shown this to be a broad neutralizing epitope as well. And it's shown here with this sequence of the stem helix that's recognized and is highly conserved among different coronaviruses. We're also taking similar approaches to make epitope-focused immunogens for that.
Kevin, could you summarize? I'm sorry.
Yes, I think I've got 3 slides and then I'm done. And then lastly, as Ralph Berg mentioned this morning, we're using chimeric spikes as a way to broaden the response. We had mRNA vaccines that were published in the paper in Science. We've now taken that same approach and we've adapted that to now make nanoparticles that can be protein-based as a way of, again, broadening the antibody response that can be generated. And so what I mean to summarize by showing you this data today are the implications for pan-coronavirus vaccines. Our design goals include integrating each of the aforementioned concepts. So in brief, we believe we're going to have to combine T-cell immunogens with antibody immunogens to activate multiple arms of the immune system. We're designing antibody-based immunogens that focus antibodies to conserve epitopes. And we're trying to broaden the reactivity by increasing the epitope diversity present during vaccination with heterologous prime boost regimens. I'll end here by thanking all of the many collaborators from many different places, Duke, UPenn, UNC, as well as our NIH funders. And I'll hand the screen back over. Thanks.
All right, thank you, Kevin. Our next speaker is Kayvon Mojarrad from Walter Reed.
Thanks very much for giving me the opportunity to talk about our approach. I was asked to give an update on what I had spoken about during the consultation back in January. So this will be very brief and hopefully we'll give some time back for discussion. Next slide, please. So what I discussed last time, just as a summary, was all the design, discovery, and early development work that we had done since January of 2020 on a SARS-CoV-2 ferritin nanoparticle vaccine based on the original Wuhan 1 strain, looking at the immunogenicity and the efficacy in multiple animal models. This is done in combination with a proprietary adjuvant from the Army as well. You can see on the right there, called ALF-Q, Army liposomal formulation. It's a unilamellar liposomal adjuvant with the sapin and QS21, and there in the center, you see negative stain EM of our nanoparticle displaying 8 trimeric SARS-CoV-2 spikes, and then the sort of globular images you see there are the adjuvant. Next slide, please. This has been covered a little bit, I think, before. Kevin mentioned it just briefly, and then I've talked about it before, that ferritin has unique advantages in being and serving as a platform or a scaffold for presentation, multimeric presentation of antigens. On the left here, you see ferritin as a monomer, and then it self-assembles into this 24-axis oligomer. Ignore the ED3 there. That was from another approach that we've taken for flaviviruses, but the idea is that you can take the ferritin monomer, add in a linker. We use a 6-amino acid linker, and then add in your antigen of choice. What we did in our approach, we had a extensive design cycle. I presented last time about 215 different designs in different categories, and we took a number of these forward, some being RBD based, some being S1 subunit, and then finally what we down selected for taking into human trials was the prefusion stabilized S2P version of the full-length spike protein that you can see on the right, with some additional mutations that we introduced in the lower hinge regions to ensure native presentation and native conformation of the spike protein on ferritin, which also offers the advantage of having this threefold axis in which trimeric proteins can be displayed in their native conformation. Next slide, please. And so this sort of boils down in terms of our animal studies. We looked at our immunogen in multiple strains of mice, hamsters, non-human primates, even nurse sharks and horses. And here on the right is kind of just a very loose graphic of the potency of our immunogen in rhesus macaques as compared to some of the other major vaccines that were out there. I add the caveat, of course, that these assays were done on different platforms and different laboratories and different studies. So they can't make a direct one-to-one comparison. However, and I'll bring this up in the context of the clinical study we're doing, we are doing immunogenicity analyses in the same laboratories and on the same platforms that were done for the federally funded, US federally funded vaccine candidates. So that allows us to do more direct comparisons with some of the other vaccine platforms. Next slide, please. What we have on the docket for our next set of animal studies, we have multivalent formulations, both a combination or a mixture of different nanoparticles, where each nanoparticle has a single, it's an immunogen displayed multiple times, and this is either RBD or full-length spike of different coronaviruses, both human and zoonotic. We also have heterologous prime boost studies ongoing in non-human primate studies with other vaccine candidates to mimic the real world situation in which we're dealing with a prime population. And if we were to boost with our platform, what would that look like? And then doing a Omicron challenge study. We've started mRNA formulations of our nanoparticle candidates and looking at this in mouse studies. And then what we're also doing is passive transfer studies of human IgG purified from individuals in the clinical trial who received our ferritin nanoparticle vaccine and challenging them with either SARS-2 variants or SARS-1. And then doing different combinations of our different immunogen designs and different adjuvants, not just ALF-Q, and starting those in non-human primates. Next slide, please. One of the things that's happened, I think, since last time that I spoke in this forum was that the Vaccine Research Center that had used ferritin as the basis for the universal flu program originally, they published a suite of studies. Here is one of them where they used the ferritin nanoparticle displaying the hemagglutinin for influenza H2 in adults who are naive to H2, and what they saw was very potent homotypic response against H2, but also pretty broad responses as well within the group of same group of viruses as H2. Next slide. And so this will be really what we're analyzing right now and have completed is our phase 1 clinical trial, which is the only other clinical trial that has been done so far with ferritin, other than the flu program at the VRC. And the way we designed it was a dose escalating study where we were evaluating 25 micrograms of our immunogen. That's the whole immunogen, so the ferritin plus the spike, and with the ALF-Q adjuvant. And then the second arm was 50 micrograms. A third arm was 50 micrograms as well, but with one less dose. So the dosing schedule is at 0, 1 month, and 6 months. However, we ran into some difficulties in terms of recruitment of naive subjects, which was the basis for our eligibility criteria, because we started this trial in April of last year at the time when many people were starting to get the emergency authorized vaccines. Next slide. And so what you can see here is that we were able to enroll a total of 29 individuals from the first 2 arms. The first arm was fully enrolled. 24 of those individuals received active product, 5 received placebo. And so this is a very small study, but what we're seeing is that we're getting very good indications of what kind of immunogenicity we're seeing against the different variants, and what kind of questions we need to be asking in a follow-up study that we're planning with an industry partner. Next slide, please.
Okay, Kayvon, can you sum up?
This is summing it up. So what we're doing is looking at reactogenicity, and then, as I said, the primary immunogenicity endpoints are from all of those that are from the federally funded vaccine laboratories, looking at binding antibodies, pseudovirus neutralization, cell-mediated immunity, as well as broader Sarbecovirus neutralization and binding, and a passive transfer in hamsters. And the last slide here is just an acknowledgement of all the different groups and individuals who are involved. Particularly, I want to highlight Dr. Gordon Joyce, my co-inventor on the vaccine. Thank you.
Okay, thank you. So our next speaker is Séverine Colléon from INSERM in France.
Stanley is going to be Professor Yves Levy, I think.
Yes.
I'm sorry.
Thank you. No problem. Thank you, Stanley.
Our problem. The person who will make the presentation is Professor Il Levy from INSERM.
Oh, okay.
No problem. Thank you, Stanley. May I have the first slide, please? So thank you for the invitation. This is, I try to follow up what I have presented in the last meeting in January. So to the design of the Pan-Sarbecovirus vaccine. Next slide. The next slide shows you the prototype of the vaccine. This is first, it's a protein vaccine, and we want to design a boost, and this protein is composed of a monoclonal antibody targeting a receptor at the surface of the dendritic cells, which is a CD40 receptor. And this antibody is always the same, it's fully humanized antibody at the VH-VL region and the FC region. And you see in the different construct that this antibody is fused directly to some sequencers. We have identified by bioinformatic approach to be conserved, highly conserved across the different sarbecoviruses. So in the last meeting, we have shown that these sequencers, around 300 amino acids for LBD or 135 for N sequence, are highly conserved around 95% to 100% across 38 sarbecoviruses, including the variants of concern of SARS-CoV-2. So the goal of this vaccine is to induce boost responses, T and B cell responses, because these sequences are rich in T cell epitope. And we have shown that this vaccine can recall in vitro some T cell responses and B cell responses and T cell, fully functional T cell, including cytotoxic killer cells. The goal is to use this in clinic with or without adjuvant, and to extend the breadth of the responses in people already immunized or already exposed to the CoV-2. So we have two vaccines under manufacturing. On the left, you see the Lonza manufacturing and the second one in France. Next slide. So just to remind some of the data we have shown last time, this is in key 18 mice model. They received 2 boosts of the CD40 vaccine with an adjuvant here, a TLR3 adjuvant agonist, Iltenol. And after that, the mice were challenged with the Wuhan strain. You see on the bottom left that the survival in the control group was around 30% compared to 100% in the vaccinated group. You see in the center, the level of the binding antibody against LBD after 2 vaccine, VV, 2 vaccine, and after the challenge in the vaccine group on the top. And you see, as expected, that in the control group receiving the adjuvant alone, we saw an increase of the antibody level only after the challenge, as expected, but this level is lower than the level of the antibody after the vaccination. On the top right, you see the neutralizing antibody activities of this antibody across the different variants. And in gray is the control group, and in blue is the vaccinated group before the challenge or after the challenge. And you see that we got a very nice increase of cross-neutralizing activities against the different variants tested. In the bottom of the slide, we looked at the cross-neutralizing activity against CoV-1 because some of the sequence of the RBD is conserved with CoV-2 and CoV-1 and SARS-1. And you see that we got also neutralizing activities after vaccination or after the challenge, even if this lower, the level is lower than the CoV-2. Next slide, please. So what we looked after that is to compare the CD40 vaccine with the Pfizer vaccine. In these animals, they received 2 boosts of the CD40 vaccine plus the adjuvant compared to 2 boosts of the Pfizer vaccine. You see the IgG binding level after the vaccination, 1 week after the vaccination, 1 and 2 weeks after the vaccination. In red, the level of the antibody in the Pfizer group, and in blue is a CD40 vaccine. And you see the level of the antibody, the binding across the different variants. In the right, this is exactly the same data, but showing the fold drop of the binding activity against the different variants. For the Pfizer, the drop is around 1.5 to 6-fold. And in the DC targeting vaccine is around 1 to 2.6-fold. So it's similar activities and cross-neutralizing activities for the two vaccines. Next slide, please. After that, we move to non-human primate studies, and you see on the left the group of the studies in the naive, the naive group in gray, the naive who received only one shot of the CD vaccine with CD40 vaccine without adjuvant, the convalescent animals, which are animals infected 9 months ago, and after recovery, they received either the PBS or a vaccine in purple, only one shot of CD40 RBD vaccine without adjuvant, and compared to 2 Pfizer vaccine in naive animals. So you see the binding here, the binding antibody against the Wuhan strain on the top or the Delta, and we have done this for different variants, just for a matter of short presentation. I showed you only the 2 viruses. You see that the naive animals, which received only 1 boost of the vaccine, they have a nice increase after 2 or 4 weeks of the antibody levels. And this antibody levels is higher than the convalescent animals here in blue. And you see in purple that the convalescent animal which received only 1 boost of the vaccine without adjuvant, they have a very nice increase of the antibody, which is stable. And this increase is at least at the same level that the animals which received the 2 Pfizer boosts. Next slide. So this shows you the binding antibody against RBD on the left and the neutralizing activities on the right. So what we can see here is the binding against the different variants, and you see that in the yellow, the animals which received the 2 Pfizer, and the level of the antibody is exactly the same as the convalescent animals which received 1 boost of the vaccine against without adjuvant. And you see in the center that the animals which received only 1 boost, the convalescent animals which received only 1 boost of the CD40 without adjuvant, they exhibited the same level of the antibody responses as compared to the natural immunity in convalescent animals. On the right, this is the inhibition, the neutralizing activities of these responses. And you see once again that the convalescent animals, which received only 1 boost of the vaccine, CD40 vaccine without adjuvant, they have exactly the same neutralizing activities as the animals which received 2 Pfizer boosts. And we extended the breadth in the naive animal only with 1 shot compared to the natural immunity in purple. Next slide. So in conclusion, we have developed a DC targeting protein vaccine which could be used with or without adjuvant, but we want to develop this in clinical at the end of this year as a boost. It induced robust binding and cross-neutralizing responses against the different variants of SARS-CoV-2, also SARS-CoV-1, in 2 preclinical models. These responses are equivalent to mRNA COVID-19 vaccine in terms of magnitude and breadth of antibody responses. And one of the advantages beyond this equivalent effect of the CD40 vaccine is that we have shown last meeting, and this is published in bioRxiv, that this vaccine, since it contains the end sequence from the CoV-2, induced also cross-reactive T-cell responses and functional T-cell responses. So we are developing and manufacturing 2 generations of DC vaccine to move in clinical trial at the end of this year, either to boost or to extend the responses against the variants, but also for preparedness against the emergence of new variants and future Sarbecoviruses. I would say also what one of the advantages of this protein vaccine is we have lyophilized vials and product, which could be very useful to avoid any cold chain issues. Thank you for your attention. I just would like in the next slide to acknowledge my colleagues. First of all, for the coordination of the mice study, it is with Véronique Godot. For the antibody and T-cell responses, Mathieu Sureno, Séverin Colléon, and Aurélie Weidman and Sylvain Cardinot. And for the non-human primate studies, my friend and a very important facility in France at the CEA, Romain Marlin and Roger Legrand. And thanks to all the people from the Vaccine Research Institute. Thank you for your attention.
Okay, merci bien, Yves. So our next speaker is move on to Dr. Lu from Fudan University in China.
Sandy, apologies. Dr. Lulu will not be able to present, but instead Dr. Shibo Yan has kindly agreed to make the presentation. Apologies,
Okay.
it's the last minute change.
Dr. Zhang?
Yes, thank you. Thanks, Danny. Because Lulu now is working in the emergency room, so it's so I have to speak for him. And actually, we are in the same group. So today we are present our strategy to use non-change against the change, the strategy to develop Pan-Sarbecovirus vaccine. That means we use the original virus strains RBD plus the human IgG FC and the adjuvant to produce antibody against all the Sarbecovirus. Next, please. Next, please. Oh, okay. This is the spike protein which contain the S1 subunit contain the NTD and RBD. So in the 2004, our group is first in the world to showing that RBD contains 6 conformation epitope that can induce the most potent neutral antibody. Therefore, RBD is the most important target for the development of vaccine and neutral antibodies. So in 2003, most other companies to develop vaccine use the spike protein, where we are used different, use S1 subunit and RBD. S1 unit actually contain NTD and RBD. Then we added a human IgG. Why we had a human IgG FC? Because we wanted to increase its half-life and allow them to form dimer and have the adjuvant effect because it can bind into the FC receptor on the antigen presenting cells, and it can easily for purification use protein A and protein G. So the result very surprised that the RBD FC elicit antibody response is about the 225 fold is much higher than the S1 subunit. Actually, S1 subunit contain RBD and NTD. And also we use a pseudovirus neutralize. We found that most cells can have the 13,000 NTD until 50s. And the rapid antibodies titers can reach at 95,000. This is the most potent neutral antibody at that time in the 2004. So this also we test the live virus. Also the most series of neutral titer is more than 10,000 and the rapid series is more than 15,000. So after that, we published more than 100 papers and to showing that the neutralized immunogenicity, the best one is RBD FC dimer, then RBD trimer, RBD monomer, and S1 proteins, S protein, and the viral particle. So the first generation of COVID-19 vaccine now we are used in the clinicals based on S protein and viral particles. That's why their neutral antibody is not high. It's just like this. You can see that, and this is the first generation antibodies. So in the mouse, most like highest is 1,500, and in the monkey and the humans is several hundred to 20s. However, you can see that RBD-based vaccine in the mouse it can reach 14,000, and the monkey is more than 10,000. That means the RBD-based vaccine induce antibody much higher than S protein vaccine. So in 2001, 2002, 2020s, so we also use the RBD-FC plus the RNA to generate a vaccine, and you can neutralize antibodies for the pseudovirus more than 8,000, and the live virus also 8,000. And also we found that this antibody can cross-neutralize SARS coronavirus and the SARS-related coronavirus from bats. So that means it's against the other Sarbecovirus. So the adjuvant is very important, but so far most of the use are RNA. So in 2 years ago, we published a paper in Science. We found that the CGAM is a STING agonist. They can make the regular seasonal flu become a pan flu because they increase both the cell immunity and human immunity against the flu virus. So we are also trying to identify the new sting organisms. So finally, we identified one is called CF501. So this is the most potent adjuvant. It's better than other adjuvant we have tested. So now we use it to immunize the monkeys. You can see the monkey can keep all the neutral antibody. So it's more than 190 days. So the neutral titer is always more than 10,000 or even 10,000. And it's about more than 24 better than animal virus. And also we test like all the SARS-CoV-2 variant, including alpha, beta, gamma, delta, and also found that also we have tested 41 nature mutant. You can see CF501 RBD vaccine, they can induce antibodies more than 10,000 or even 10,000. But where is the animal is much lower. So that means that also this is have induced a very potent and broad neutral antibodies against all this SARS-CoV-2 variant we have tested. And also recently we tested the Omicron and also found that this monkey series is very effective against monkey errors. Even in the day 190s after immunization, that means they still have a neutral titer is more than 240s. And even it's much better than animal. Animal is only 6.8. So this is a summary. CF501 RBD-FC based Pan-Sarbecovirus vaccine is more than 24 more potent than animal RBD based vaccine. So it is effective against SARS-CoV-2 and all its variant of alpha, beta, gamma, delta, and Omicron. And also neutral antibodies more than 10,000. And also against the SARS-CoV-2 and its variant. And also against several of SARS-related coronavirus from bat. So this is our result. Thank you for your attention.
Well, thank you, Dr. John, and for bringing up the importance of adjuvants to immunogenicity. All right, so our last speaker in this session is Pamela Bjorkman from the California Institute of Technology.
Thank you. Dr. John, could you please stop sharing your screen? Thank you.
May I share my own slides?
Yes, you can go ahead.
Okay, thank you. So thanks for inviting me. I wanted to talk about our work on a mosaic nanoparticle approach. And so I want to start by saying that we've been trying to do this for HIV and flu and Zika virus and so on for 4 or 5 years now, based on this hypothesis for how you might get better cross-reactive antibodies in the case of a virus that has conserved and variable epitopes. So let's say that you've made a nanoparticle and it presents multiple copies of an antigen that has strain-specific distracting epitopes that are symbolized by these orange triangles here. So now what's going to happen when you go into a germinal center and you trigger a B cell with B cell receptors that have bivalent membrane-bound antibodies is that you will cross-link a lot of these orange type of receptors that see this very accessible strain-specific epitope. But now let's say that instead what you did is you injected a mosaic nanoparticle, and now if you've got a randomly arranged mosaic nanoparticle where no 2 strain-specific distracting epitopes are next to each other on average, you can see there's another orange one here, but if you put enough of these RBDs on, it's not likely to be next to each other. So then you're not as likely to make these orange antibodies, which is what we do not want. Instead, what we want are these purple antibodies to be stimulated, to be produced, and these are against the more conserved parts of the antigen. So what are these on Sarbecovirus coronavirus spike protein? And so on the left there, you're seeing the spike trimer structure as solved, for example, by David Wiesler's lab, and you're seeing an RBD in a so-called up position where it can interact with ACE2. And from our structural studies, mostly done by Christopher Barnes now professor at Stanford, then a postdoc in my lab, we looked at the conserved and variable parts of the RBD by plotting the sequences of Sarbecoviruses and variants of interest and variants of concern onto the RBD surface. So you're seeing that from two sides now, and you can see that at the top, you have these, what we call class 1 and class 2 epitopes, which overlap with the ACE2 binding footprint directly. You make very potent antibodies to these, but unfortunately they vary between Sarbecoviruses and they vary in the variants of concern as well. So what we're really aiming for are the more conserved epitopes down there, the class 3 and class 4 that are mostly purple. And so this is a movie showing that the purple part of the RBD epitope is actually less accessible than the orange part, partly explaining why you tend to get those immunodominant epitopes against the accessible variable epitopes. So now we're going to plot the orange and purple surfaces onto that RBD. So you can see it's a little bit hard to get the antibodies in to access that purple region, but of course you can do that. And so we're going to show you some structures that our lab has solved of antibodies from human donors that do target that purple region, but they also, through steric hindrance, block ACE2 binding. So we call these combination class 1, class 4 antibodies. Okay, so that's what we're trying to do. So when the pandemic began, Alex Cohen, then a graduate student in my lab, now a postdoc, combined, he made different types of nanoparticles using a plug and display approach that involves spy catcher spy tag technology. So we're using an RBD that's shown there we're using a nanoparticle that's shown there schematically on the right. It's related to the one that Neil King's lab developed, but this has the spy catcher particle on it. So we spy tag the RBDs, we incubate them, and they bind covalently. So Alex chose a collection of 8 receptor binding domains from Sarbecoviruses, the potential pandemic concerns, so SARS-CoV-2 plus 7 animal coronaviruses. And then he made mosaic 4A, 4B, and then mosaic 8. And the important point here is that we're going to look for, as has been described, responses against what we'll call matched strains, and those have an RBD on the particle, versus mismatched. So these are a collection of some of the mismatched strains we're looking at. Importantly, these include a number of class 3 epitopes. And now you're seeing what that a computer model of what that nanoparticle would look like. And the important point here is that we do not want RBDs to be adjacent to each other that are the same. Okay, so here's a summary of Alex's published paper and then some more recent work at BioQual with vaccinated non-human primates. So these were vaccinated with our mosaic 8 nanoparticle. And so as you can see in both mice and non-human primates, we get a better response. In the mice, you can see we get better binding responses using the nanoparticle across the Sarbecoviruses if we use the mosaic compared to the homotypic nanoparticle, which is only SARS-CoV-2. And then in the non-human primates, we're just comparing the mosaic 8 to unimmunized. And then we're getting cross-reactive neutralizing activity here. The mismatched strains are listed there with the blue rectangle. So here what I want to do is quickly summarize the non-human primate data. These non-human primates were vaccinated once and then boosted twice and then challenged with either SARS-1 or SARS-2 Delta. Both of these would be mismatched because the SARS-2 that was on the particle was beta. Okay, so the point of this ELISA as a function of time is just that we're not seeing differences in binding responses across different SARS-2 variants, and Omicron's in there, but you can't tell which one it is because it doesn't matter. But we're also seeing good responses against a variety of mismatched viruses, including some in clades 3 and clades 4. And now this is neutralization data. And again, we're seeing cross-reactive neutralization from the vaccinated animals across these mismatched clades, including clade 3, the BTKY72 represented there, and then some authentic virus neutralization that was done at BioQual against SARS-2 and its variants. And you can see we're getting really good responses against Omicron. So then as far as the protection goes against both SARS-2 and SARS-1, we're seeing protection with the mosaic 8 immunized animals. This is compared to the control group, which did not receive the immunization. And you can see that in all cases in bronchial alveolar lavage for SARS-1 and SARS-2, the vaccinated animals are undetectable in terms of infectious virus titers. And we're seeing a little bit of SARS-2 in the upper respiratory tract here, but it's reduced by a factor of 1 or 2 logs. And so this would be similar to vaccines in humans where you do see some infection in the upper respiratory tract. But so far this looks good, like protection. So then we did challenges in the lethal K18 human ACE2 transgenic model, and these were done by Neeltje van Dormelaan and Vincent Munster at the Rocky Mountain National Labs. So here we're using the mice to then challenge with either SARS-2, which is a matched challenge, or SARS-1, which is a mismatched challenge. And in this case, we could do more cohorts. So if you compare, we have 2 types of mosaic 8. We have the original mosaic 8, and then we have one that's modified with glycans to attempt to do immunofocusing to get the epitope, to get the antibodies to stop focusing on the class 1 and class 2 epitopes that overlap with the ACE2 binding site. So this was our attempted vaccine design and of the RBDs. And so what you can see for the SARS-2 challenge is the animals immunized with the mosaic 8 or the homotypic SARS-2 nanoparticles were completely protected against weight loss. And you can see from the survival curves that they were also completely protected. That goes out to 28 days. And one of the animals in the mosaic 8 GM group died, and it lost, there was an average weight loss, but there was some recovery there. So I think that we want to explore this further, but we're wondering if these attempts to immunofocus away from these epitopes might not be such a good idea. However, in the case of the SARS-1 challenge, the only protected animals were the mosaic 8 or the mosaic 8 GM, shown there by the thick blue and green lines. There was no weight loss, and there was complete survival out to 28 days. Okay, so we wanted to understand the mechanism of the protection in this case. And so Jesse Bloom's lab has been doing total RBD mapping, mutational mapping. So they have a yeast library that presents every variant of the RBD residues. So you change them all to every other residue, and the ones that can be expressed on the surface of yeast, you then screen those with, for example, a polyclonal antiserum. And that's what was done here with 6 different sera from mosaic 8 immunized mice or homotypic immunized mice. And then they look at which sites affect the binding. And it's really clear here that the mosaic 8 is targeting a different place on the RBD than the homotypic. So if you look at the bottom, the homotypic is largely focusing on that class 2 residue 484, which is the site that's induced by natural infection and by vaccination quite strongly, but that is a residue that changes in the variants, whereas the mosaic 8 is actually inducing the site that we wanted. This is on the purple regions here. Those are mainly class 3 and class 4 epitopes. So of course, now the question I always get is, well, if you start targeting the conserved region, the purple region, what's going to happen if you made a vaccine like this? Aren't those regions going to mutate? And we don't think so. And we think this is shown by natural evolution of Sarbecovirus spike proteins, because remember, the RBDs have to adopt both up and down positions. And if you look at what the RBD looks like when it's down, and you map the contact regions of the down RBD with the rest of the spike trimer, they very closely match to the purple regions here. So we think that we've seen natural evolution in process where animal Sarbecoviruses don't tend to change those residues, and the variants of concern also don't. So we're hoping that by focusing on this purple region here and making this mosaic 8 vaccine, we could make a vaccine that could be put into humans that would never have to be changed to protect against future spillover events from animal Sarbecoviruses. So I want to quickly thank everyone. I haven't had a chance to really talk about what most of them did, but here's their pictures and the acknowledgements. So thanks for your time.
Thank you very much. So I will now ask Dr. Phil Krause to speak about the evaluation of these new approaches.
Thanks, Stanley. What I'm going to present here is a draft framework for the evaluation of new vaccines, which has already been discussed at a number of other WHO meetings. But interestingly, the same approaches that we might use to think about other new COVID vaccines also can be used to think about Pan-Sarbecovirus vaccines. Because at least in the simplest case, of course, if a Pan-Sarbecovirus vaccine would meet our current needs for SARS-CoV-2, the things that the regulators have already told us will be okay for that should also be good for a Pan-Sarbecovirus vaccine. And of course, there'll be a discussion later on in the meeting about perhaps other things that can be said about Pan-Sarbecovirus vaccines. And it may also be that as we've thought about how to do this for new vaccines for SARS-CoV-2, there's some insight that can be applied to the Pan-Sarbecovirus situation as well. So next slide. The goals, of course, that motivate this draft framework is the desire to increase supply of vaccines that are likely to meet the TPP criteria. And there, of course, those criteria, again, for effectiveness against severe disease are the most important ones. And I posit that that's probably also the most important thing for a Pan-Sarbecovirus vaccine, if we want to prevent severe disease. We'd like new vaccines against SARS-CoV-2 to be as variant resistant as possible, unless they confer major offsetting advantages. And the question that had come up of whether or not we can use what we know about the immune responses to these vaccines to consider a new vaccine for EUL based on an already authorized comparator. And the answer was that probably under some circumstances one could do that. That's what I'm going to go over here. When those circumstances aren't met, one would need additional data prior to EUL. And in any event, as we talk about how we're going to demonstrate effectiveness of vaccines, we're assuming that safety has already been addressed. And so it's not that we're taking safety off the table, but safety, of course, is a critical thing. But the challenge, the scientific challenge here, perhaps, is figuring out how to demonstrate effectiveness, not how to demonstrate safety. Next slide. So there are a few considerations that motivated the thinking behind this. And many of these have already been covered. Neutralizing immune responses appear to mediate protection, and levels of neutralizing immune responses can predict protection, not prediction, against symptomatic disease. What we've seen with the existing vaccines, though, is that after neutralizing antibodies wane, there are other responses that can take over and still maintain protection against severe disease. So if there are very high neutralizing antibody levels, then you can still be protected against both symptomatic and severe disease. But as they wane, these other responses become more important. And the other thing that we've learned, I think, over time, as the new variants have come in, is that these non-neutralizing responses appear to play a greater role in vaccine protection against variants. And the reason is simple. It's because the variants have evolved mutations, in many cases, in the specific domains where the neutralizing responses are effective. And so the immune system relies on other domains in order to provide protection against the new variants. And these non-neutralizing protective responses, as been discussed many times, can include cell-mediated responses, in which one might include T cells and memory B cells, but also FC-dependent non-neutralizing immune responses. But I think, as several of the speakers in the last session said, ideally new vaccines for Pan-Sarbecoviruses as well as for SARS-CoV-2 would induce both neutralizing and non-neutralizing protective responses. Next slide. So the framework that is being presented then provides an approach to considering vaccines based on information about the mechanism of action of a new vaccine and immune responses relative to those of an already authorized comparator. And as I mentioned, this is really, the thinking here is intended to apply to all SARS-CoV-2 vaccine situations, whether these are new vaccines, whether these are booster vaccines, whether these are variant-specific vaccines, or indeed if these are Pan-Sarbecovirus vaccines. Next slide. The framework that provides an alternative for placebo-controlled clinical trials under certain circumstances. Now, this is not exactly the same as a serological correlative protection, because what we're doing is we're using more information than what we would have if we were using a serological correlative protection. And because of that, we need to think very hard about what is the comparator and what do we know about the comparator, because we're really comparing the immune response of one vaccine against another one where the existing vaccine is known to be effective against what we want, which is a severe disease against caused by currently circulating variants. And so that choice of the comparator has to be well justified. Next slide. So the key questions then come down to what's the breadth of antigenic composition relative to the proposed comparator? To what degree is the likely non-neutralizing or mostly CMI response using the new vaccine likely to be similarly proportional to the humoral response against the comparator vaccine? In other words, if there is a good humoral response, is that going to also predict these non-neutralizing, if there's a good neutralizing response, will that also predict the non-neutralizing responses equally well for the comparator as well as the new vaccine? And then surely it matters how good the comparator is. A comparator which is highly effective may be viewed differently than a comparator which is less effective, and we would want a new vaccine to perform obviously as well as a vaccine that is also performing well. And I'll just point out that this breadth of antigenic composition has come up several times, and to be interested in what people at this meeting have to think about this is that there's an obvious situation that if a new vaccine has less viral sequence than a comparator, then it's going to present fewer important cellular or non-neutralizing humoral epitopes. And so if it is presenting fewer of these epitopes, then it may be that it's starting with a strike against it, because it will be less likely to induce the same breadth of non-neutralizing protective responses. And of course, I've mentioned, already explained why it is that the cell-mediated and these non-neutralizing protective responses are important, especially as we're thinking about variants and especially as we're thinking about duration of protection. Next slide. So to go into this in a little more detail, and some of this I've already had. So if we think about the effectiveness of the comparator, we care about the effectiveness against severe disease caused by circulating variants. And so it's critical to be able to make direct or indirect comparisons of the immune responses induced by a new vaccine with those that are induced by other vaccines of known effectiveness. But of course, there may be some concerns about how well we understand the effectiveness of those known vaccines. And these are things that can be considered in this framework. But this would need to be the most current data available about vaccine effectiveness against severe disease caused by these circulating variants. And the degree of effectiveness of that comparator should affect the standard that a new vaccine would be expected to meet. Next slide. And then the second question is whether the predicted or likely CMI response using the new vaccine is likely to be similarly proportional to the humoral response against the comparator vaccine, which is another way of saying is if you get similar humoral responses or similar neutralizing responses, is that going to predict all of these other responses? And one could argue that if they use the same platform, they're highly likely to predict these other responses. But if the platforms are different, then you would want to have some additional data to show that these non-neutralizing protective responses of the new vaccine will be at least as strong as those of the comparator vaccine. Next slide. And then the question is, what's the breadth of antigenic composition relative to the proposed comparator? And if the breadth is, as mentioned, if it's lower for the new vaccine than the comparator, it's likely that it won't induce responses to as many cell-mediated and non-neutralizing humoral epitopes. And so one would then like to have clear data that indicated considerably stronger or more durable neutralizing responses, or otherwise one might be concerned that the new vaccine would be less resilient to waning of neutralizing responses and less resilient to new variants. So there might be a presumption against immunobridging to a comparator with broader antigenic composition unless there were additional data to support that. Next slide. So this is then all put together in a table, which has been presented at previous meetings, that basically present this possible framework for evaluating new vaccines. And it's based on scenarios that consider the effectiveness of the comparator against severe disease caused by circulating variants and the likelihood that humoral responses to a new vaccine will predict cellular responses. And then this could then lead to thinking about how a new vaccine would be evaluated. Next slide. So which shows this table, and I'll just go into a little bit more detail here. So the three questions that I've already been over are listed as questions 1, 2, and 3. So depending on what the answers to these questions are, you might find yourself in one of these 5 scenarios here. And then depending on which of these scenarios one found oneself in, one would go to this green line and answer the question of what additional data one might need to authorize the new vaccine. And so next slide, I'm just going to focus in on scenarios 1 through 3. In scenarios 1 through 3, we're contemplating potential immunobridging where there's a high likelihood that the neutralizing immune response to the new vaccine will also predict other protective immune responses relative to the comparator, where the only difference between these is where the comparator might have different effectiveness in preventing severe disease caused by circulating variants. And so if it's highly effective, we're in scenario 1. If it's modestly effective, we're in scenario 2. If it's not very effective, but it's still EUL listed, then we might be in scenario 3. And the idea is that if the comparator is highly effective, it should be okay to study your new vaccine using non-inferiority criteria. But if the comparator is less effective, one should use superiority criteria to show that the new vaccine actually is inducing a better immune response than the previous one did. And this is, of course, an immune response to the circulating variants that we're concerned about protecting ourselves against. Next slide. Scenarios, right, and so this just says what I just said. Next slide. So scenario 4 and 5 contemplate situations where, and this is more detail that I'll leave in the slides. These slides will all be made available later, but it's more detail than I need to go through right now. So scenarios 4 and 5, next slide, contemplate situations where in scenario 4, the neutralizing response is good, but there's some concern about the non-neutralizing responses because we don't think that the neutralizing responses are as likely to predict the non-neutralizing protective responses. And if that's the case, we'll want to have some additional data. And then in scenario 5, next slide, we have a situation where there's something about this vaccine which makes us think that it's likely to be that where the neutralizing response is not as good, but there's some reason to think that there's a very good cell-mediated or mucosal response to this new vaccine. And so in scenarios 4 and 5, there might be some concern about this immunobridging approach based only on neutralizing antibodies. And in these cases, one would look towards gaining additional clinical data before one would make these vaccines broadly available. And that data could come from human challenge studies if a suitable challenge strain were available, or from in-deployment studies or effectiveness against severe disease, with the idea that instead of simply giving vaccine to people in deployment in the order in which they signed up, it would be randomly allocated to people who are eligible for vaccine, based on the idea that the vaccine might have some limited availability anyway, and that those who received vaccine early could be compared with people who received vaccine later. And this has been something that has also been discussed at previous WHO meetings and is seen as a possibly very reasonable way of obtaining randomized effectiveness data very, very quickly against for new vaccines. Next slide. So that's the end of this slideshow. And so what I hope this does is it helps set a scene which makes us start thinking about how it is that not only we can make Pan-Sarbecovirus vaccines, but how we can use the thinking that's already gone into evaluating SARS-CoV-2 vaccines, including those that might address new variants, and in order to speed our thinking about how to evaluate them. And of course, we'll hear next from the manufacturers, and I'm sure that one of the things they will care most about also is how it is that they will be able to support their claims about these vaccines. So be very interested in hearing their ideas on this topic as well. So thank you very much, Stanley.
Well, thank you, Phil. And this is crucial because we're in principle going to make these vaccines before there is a new variant that eludes our vaccines. So this is very important for the future. I will now turn over the podium, so to speak, to Marie-Paul Kini to conduct a discussion by vaccine developers.
Thanks a lot, Stanley, and thanks, Phil, because what you have presented should really give food for thought for this session, this vaccine developers panel. So we will have 4 vaccine developers: Janssen, Serum Institute of India, Osivax, and Zis Salak. So the way we will handle this panel is that we will first have each of the manufacturers present for just 3 minutes in order for everybody to know what is their vaccine that they know intend to get to registration, so that we understand the difficulty that they might face in terms of Pan-Sarbecovirus vaccine development. And then we will focus on 2 questions. The first one, as stated in the agenda, is the following: what are the challenges you are experiencing with your candidate vaccine? And this is indeed should be understood, especially in terms of demonstrated effectiveness or efficacy and registration. And the second question is about what the international research community could do to facilitate the development of Pan-Sarbecovirus vaccines, and your vaccine in particular. So if we may have start in the order that I have in my agenda here, I would like to start with Janssen. So who will be presenting for Janssen? Can you put your slide on? Hello, Hanneke.
Hi, Marie-Paul. It's Hanneke Schuitenmaker presenting.
A pleasure.
Hi, I will share my screen.
Please go and share your slides. So, what I'd like to have is a short presentation on the principle of your Pan-Sarbecovirus vaccine, so that we understand what is it, which makes it different from the one that you have already registered, and hear mainly about your thoughts about how you want to bring this to registration. Over to you then.
Yes, thank you, Marie-Paul. Let's go. What we have done till now is really looking for the identity in sequences between different members of this Sarbecovirus family, and also compared it with the identity between the current variants of SARS-CoV-2 to really see how distant it is. And when we do that analysis, we see that while the sequence conservation in S1 is relatively low, that there is high sequence conservation in S2 part of spike, which could be an interesting first attempt. But if we look at that heterogeneity between variants, we see that it is much larger than what we have seen so far. Although with the Omicron variant, we of course have seen a major genetic jump with variants, and what already was emphasized by Marion Koopman, that we can even speak of a new serotype. So this is already something that can teach us on what the breadth of immunity needs to be, and how this can be accomplished. And what we have seen with the Janssen vaccine in the current pandemic is that the effectiveness, so this is real world data, that effectiveness of the vaccine against initially the Wuhan variant and then subsequently the Delta variant was preserved. Now, one could argue this is not yet a big genetic distance between these variants, but it already shows that there is some wiggle room for protection against hospitalization. And this is also when we compare it in a vaccinated versus an unvaccinated group, we see that it is preserved over time. Another important piece of information comes from the CISONKI study that is done in South Africa, which is a study in healthcare workers that were first receiving a single dose of the Janssen vaccine, And this rolled over into Sisonke 2, where part of these healthcare workers received a booster dose 6 to 10 months later. And that happened to coincide with the emergence of the Omicron variant. So this allowed us in a real world setting to see whether the vaccine that is based on Wuhan would still protect against Omicron variant. And you can see here in the table that indeed, as of 14 days after the booster, that protection against hospitalization was still conserved. And this was done in the national database, but also in the epicenter of the Omicron outbreak. We see that these percentages of vaccine effectiveness were rather consistent. Now, when we look at the immune responses elicited by the Wuhan-based vaccine, we see that neutralizing antibody titers against variants is lower than against the reference strain that is homologous to what's in the vaccine. And we see that the neutralizing antibody titers against Omicron are really low. This is true for the Janssen vaccine, but as a comparator, here is the Pfizer BNT 162b vaccine. This is data that is available on the preprint server. However, when we look for both vaccines and T-cell responses, we see that T-cell responses against variants is quite well conserved, and that responses against Omicron are as high as against the reference strain, again, for both vaccines. So this altogether may suggest that indeed the T-cell responses could be important for protection against severe disease and may allow some breadth of protection and could be a very attractive target or to target upon vaccine. Now on this slide, I listed some of the challenges, and I think a lot of it is very similar to what Phil Krause already mentioned. So I think what the discussion will be is what is an acceptable TPP? Do we need a vaccine that elicits both humoral and cellular immunity, or if we would focus on high protection against severe disease, could we aim for another immune component? Very important, and also touched on by Phil in the end, is how to demonstrate breadth and duration of vaccine efficacy in humans in absence of multiple circulating Sarbecoviruses, and of course with the unknown of what the next virus will be, and how to get a label that includes that breadth of protection. Pathway to licensure, even for EUA, is challenging, and I think this was all touched upon by Phil. So I'm not sure if animal rule was there, but from the Ebola work, we of course know that that is a potential path, human challenge models, and of course human immunobridging to currently effective vaccines to see what component could help. And another important aspect for us is whether the Pan-Sarbecovirus vaccines will provide sufficient protection in comparison to targeted vaccines that may be developed in response to new outbreaks and that are based on rapid response platforms, because that would only leave a defined period of time in which these PAN vaccines would be used. And so it really is also on what is expected, whether you need protection against infection, which will probably require strong neutralizing antibody responses, or whether protection against severe disease, hospitalization, and death by these immediately available PAN vaccines are considered sufficient. And also said by Phil, the EUA would be based on availability of a safety database, but of course, if we would switch to other platforms, unknown platforms, new adjuvants, etc., that of course remains to be seen how acceptable that is to go without a large database. And that was what I wanted to share. Thank you.
Thanks a lot, Anneke. And if we take the point of view of Janssen, actually, what are you expecting? What is it that would be the most helpful for you in order to help you develop such a Pan-Sarbecovirus?
Yeah, so we would be interested in an immunogen that gives this broad cellular immune responses. We really think that the breath is most easily obtained through cellular immunity, because we see also in the sequences that with all the variants of SARS-CoV-2 that have emerged now, that the number of epitopes for the T-cell responses that is impacted is really minimal. So we think that there is some room for variation, but also a good target to get to protective immunity.
But are you, at Janssen, are you developing vaccines also that are based on other antigens or others that could potentially be combined with what you have with you add 26 with spike, and that would induce also cellular immune response against more conserved epitopes?
Yeah, so not yet at this point. So we are looking at it, but we are still recovering from the current work. So, but there is for sure, of course, an interest in how can we be better prepared for future situations.
Thanks a lot. So we'll take the other three participants to this panel, and then Hanneke, I'd like you to come back so that we'll have a more general discussion. So the next one will be from Serum Institute of India.
Hi.
Oh,
hello,
hello Rajiv. Such a
hello.
pleasure to see you, although remote.
Yes,
Yes, very well. Thank you.
To see.
Rajiv, over to you.
Yeah, try and wind up as quickly as possible. Jimena, can you show the slide? Because you said you will be sharing the slide. Can she scroll the slides? I sent already the slides.
We can see them. Go ahead.
They said that deal this. Yeah, Jimena, thanks. Yeah. Can we go to the next one? Yes, considering the performance of almost all available vaccine and their efficacy, effectiveness against the variant, it seems logical to have a vaccine which offers something beyond RBD. Coronavirus is known for not generating long-lasting immune response, that we know. COVID-19 has something unique in delaying the interferon response. And in order to encompass all present and future coronaviruses, reactive vaccination campaigns may not be a complete solution. Next one. All these facts together pose huge challenge in getting Pan-Sarbecovirus vaccine. Next one. The logical is to generate a response mimicking the infection. That's the entire story that we will deal with. Breakthrough cases show a significant rise in neutralizing antibodies and IgA response. Can we go to the next one? Within the scope of available information, the first step to evaluate the development of Pan-Sarbecovirus vaccine is to look at response to multiple antigen and mimic the site or mode of infection. Immunizing the population with most conserved part of the virus, broader antigenic composition, and continuing the immunization right from the infant stage may be one of the strategies to block the transmission. Immunizing infants routinely will introduce the children with memory to coronavirus. Generating memory pools from the young age and boosting it at regular intervals may reduce the impact of mass spread. Prime and boost strategy with live attenuated vaccine along with inactivated vaccine may be the best alternative. It's like a flu story. Can we go to the next one? Probable solution. So one can address the issue to an extent by assessing live attenuated vaccine delivered intranasally and or injectable route and look for neutralizing antibodies, local IgA and CMI. Can we go to the next one? So what is the available platform? Next one. We are working here on a live attenuated platform, which is a codon pair de-optimization, which replaces preferred codons with synonymous codons that are translated more slowly. Virus is identical at the amino acid level, but is attenuated, and large number of silent mutation, or more than 200 mutations, is insurmountably high to back mutate. Can we go to the next one? Here we can say a surrogate challenge sort of thing. The slide is a little crowded. So we evaluated in human being 3 doses. One is low, the other one is middle, and the blue one is the high dose. And we gave a second dose and looked for shedding, which indicates that the high dose subjects do not shed the virus, which indicates that the first vaccination introduced through nasal offers them a protection by not shedding the viruses. Can we go to the next one? This is a cell-mediated immunity, which is performed at ELISpot, which is assay performed with commercially available SARS-CoV-2 peptide pools against SARS-CoV-2 SMNO, 4 viral proteins, including spike. And you can see that the high dose, which is COVID-Live 5 into 10.6 PFU per 2 doses, will give you and is giving us more than 2 to 4-fold rise when they are sensitized with this pool. So data for the day 36 time point, 1 week post second dose plotted for individuals who received placebo and participants who received 2 doses of vaccine, which this is really the key, the CMI response that we are trying to really look for. There are so many other studies in animals that we have already completed, including the hamster challenge, monkey challenge. We have also evaluated the cross neutralization with beta viruses, alpha viruses, and now in the process of evaluating the Omicron neutralization ability as well. Next one, please. Biggest hurdle is the design of clinical trial when the comparator is an injectable preparation. It is difficult to match the serum antibody response for proving non-inferiority. Can we go to the next one? There is a need for initial evaluation based on CMI, IgA, and IgG response, and then a trial design which compares the immunological effect oblique vaccine take. I think this terminology comes from flu vaccine, by combining all these responses mentioned above. I think I will stop here and can pose any question. This is a very short thing which I presented.
Thanks a lot, Rajiv. If I can ask, so what you say is that you think that a good Pan-Sarbecovirus vaccine would be made using a prime boost, taking a live vaccine first in order to elicit responses, all kinds of responses, CMI and neutralizing, and mucosal, and then come with an inactivated preparation of some sort, a different one. So what you say is that the challenge that you have would be to register only the live attenuated, because you will never get to a neutralizing antibody titer by mucosal route as high as what you get parenterally. But if your prototype is actually a prime boost, you should actually get to quite high antibody neutralizing antibody titers, no?
See, because yeah, you are right, but now we are placed in a situation where majority of population is already vaccinated or infected and vaccinated. So when we give the live vaccine, it could be a boost from that angle. And if it is a primary series, then we feel that in order to get the first immune response, I think we should go with 2 doses of priming and then continue with maybe inactivated if we find that the durability of this vaccine doesn't go beyond 2 years or 3 years. Today, we don't know the durability of this response. So it could be a solution that you have a prime and boost, and then you look for the waning of the antibody, if at all it is there, or blockade of transmission. What we have seen in most of our experiments is we get a lot of neutralizing antibodies against NP and very less against RBD. So when we compare the neutralizing antibody using only RBD in ELISA, then comparatively this nasal vaccine shows lower response. But when we compare NP, then it shows very high thing. So what we were trying to say is humoral antibody, but not only against the RBD, but maybe against NP, which many of publications shows that even infected people shows a very big boost in NP titers. So that's what we are trying to evaluate at. Maybe on longer run, if this platform safety is acceptable of codon de-optimization, then we can mix maybe a variant or maybe even SARS-1 or the other viruses, if at all we see the safety of this platform. So far, there is no issue of safety. The reversion is not an issue, and impossibility of reverting 200 mutations in the RBD zone at that time. So our main challenge is the clinical trial design.
Thanks, Rajiv. And what do you think, what would you expect from the international community besides the new framework developed by WHO and Phil Krause?
Yeah. So beyond effectiveness, if we have to see, then we have to see the take of the vaccine. I mean, you have seen that only we have seen breakthrough cases where a very high titer RBD, anti-RBD titer doesn't stop neither transmission and not infection. So if we look at the humoral antibody plus local memory, local IgA, what we do in flu usually, and then define something called as take of the vaccine. Then I think it will be easier, even if we have a comparator in form of injectable vaccine.
Thanks a lot, Rajiv. So if I still have 5 minutes, I'll take you back at the end. So the presenters, so if I may call them back, then we'll have Hanneke, Rajiv and Alexandre. And if we can have some exchange actually to see what's common between, you know, the challenges that you all face. And I would say that it's mainly about the measurement and the validation of T-cell responses as mediators of protection or effectiveness. You know, how do you see this? Do you see a common way for which that would be useful for all of these development? Although Hanneke, you are still on spike vaccine, and Alexandre is anything but spike, and Rajiv is on spike plus all the rest. So the composition is very different. But do you see anything that should be done that would help actually the three approaches?
Yeah, so for us to look at spike is really because we would benefit from the immunobridging that we could do to current vaccines, because we know what these immune responses do against SARS-CoV-2, and then see if you get similar magnitude of responses that could work against a new virus. So I think new immunogens may be very attractive, especially if you want to increase the T-cell response, but it comes with additional challenges on the regulatory pathway, I would expect.
Yes, Alexandre, I see you would like to.
Yeah, I would certainly, you know, go in the same direction as what Dr. Schultze-Macher is saying. I mean, we certainly appreciate that this creates additional burdens in terms of regulatory path. That being said, we really think that, you know, addressing a different target can really help overcome the, you know, potential barriers that we would have with, you know, one single antigen. Also appreciating that, of course, spike should be, you know, is and will remain the primary target for you know, such viruses. We really see this as an important and actually crucial way to anticipate any, you know, potential mutations on the spike that would actually, you know, really impede the efficacy of these vaccines. And I think the, you know, nature has told us in other viruses that this can clearly happen, and so that's where we're standing now. We, you know, as where we're standing, clearly, I think the regulatory pathway is an issue. We don't have this as a very short-term issue because we, you know, we're really laser focused on let's say initial clinical development, but we and I would say regarding the regulatory path, we're also looking at what's happening on, you know, let's say other viruses such as influenza, on which we're also working on, which, you know, may also let's say teach us what can be acceptable from a regulatory path. But that's I think it's an open question, but I would clearly concur with what Dr. Schultze-Macher is saying.
Thanks, Alexandre. As I told you a long time ago, you don't think only the next step, work on 4 steps ahead. So Rajiv, you know, your approach, which is based on a comprehensive response at the beginning, having a T cell and a B cell response and a mucosal immune response against a live attenuated vaccine, and then coming with what you say is an inactivated, but actually instead of having an inactivated which is not that immunogenic, you could also envisage to come with a booster using a protein, a subunit protein like you have with your Covovax vaccine, which is an adjuvant, and add an N specific antigen like developed by by Osivac. So I know it's a lot of different vaccines to put together, but how do you see then the future?
I think it may be a little adventurous to stay at this stage, but we were first trying to inject and even deliver intranasally, half the dose intranasally and half the dose inject, which will generate humoral antibodies, and also like measles, you inject and you get a plethora of antibodies to various components, and then also generate a memory as well as IgA response to block the transmission to an extent intranasally. So that is also on the card. And if this platform is acceptable, this is a codon de-optimization live attenuator. So we are going away from 20 years of developing an attenuated strain. So if this platform shows safety issues completely gone, then we can make a mixture of injectable plus nasal, or an injectable and a nasal booster from the same virus, and that is the primary thing which we are working around. But our regulatory problem is, if we have to compare with the GMC ratios of anti-RBD titer, then it is going to be difficult. Number two, we don't have a standard preparation of showing what T-cell immunity means, and what is 4-fold rise, 2-fold rise, 3-fold rise is good enough to say a protection against the comparator. And the best thing is there any marker for an IgA response which will show in the intranasal cavity, that if we show that there is a good amount of IgA produced by intranasal inoculation, then will that be an acceptable proposition for either a booster form or a primary form with 2 series? But there are no clear things so far as a standard preparation for showing even CMI as a good or bad thing.
Thanks a lot. So a lot of challenges for the design, indeed, of what we want to demonstrate, how we want to demonstrate Pan-Sarbecovirus effectiveness also, which is a big question, and associated with that, a lot of questions also on the regulatory path. So with this, I would like to thank you very much, and I would like to give the chairmanship back to Stanley. Stanley, over to you.
Well, thank you very much, Marie-Paul. That was a quite interesting discussion. So at this point, I would like to welcome Florian Kramer from Mount Sinai Hospital in New York to give us his thoughts.
Thanks
Florian?
a lot. Thanks a lot, Stanley. I'm going to share my slides here, and I'll share thoughts that I have about both variant-proof and Pan-Sarbecovirus vaccines with a few slides here. Before we go into my thoughts, I just wanted to reiterate what we are talking about when we talk about Sarbecoviruses. We have here in this phylogenetic tree marked in red the genus of beta coronaviruses, and the subgenus that is marked here as Sarbecoviruses. So you see SARS coronavirus 2 and its Omicron variant here. So the phylogenetic distance between those two is not very big. But then we actually have a whole branch here that stands for the Sarbecoviruses. There's only a few members shown here, but actually there's huge diversity in the Sarbecoviruses, and that's shown here. This paper, which is really an excellent overview that I can recommend, proposed 5 lineages here, with lineage 1 having the SARS coronavirus 1 as a member, and lineage 5 here having SARS coronavirus 2. And you can see these are geographically distributed, but there seems to be quite some diversity here. The very important thing to keep in mind is that some of these viruses use ACE2 as receptor, but the majority does not, and that complicates things. And so both SARS coronavirus 1 and SARS coronavirus 2 use ACE2, but the vast majority of Sarbecoviruses that are circulating that have been detected do actually not use ACE2. And that is really a challenge for vaccine development, I think, as well, if you talk about Pan-Sarbecovirus vaccines. Now, I think one important thing that we have to keep in mind when we talk about Pan-Sarbecovirus vaccines is, what do we want to protect from? Do we want to protect from infection? And here we really need neutralizing antibodies, and the target should likely be the receptor binding domain. Do we want to protect from disease? Again, neutralizing antibody, as has been shown, is very important here, but other immune mechanisms might contribute as well, including antibody effector functions or cellular immunity. The target here is likely also the RBD or the spike in general. Or do we want to protect from severe disease? And here it's a little bit different, because here binding antibody could contribute a lot through effector functions. The anamnestic response through T cells and memory B cells becomes very important, because there's more time for that response to really kick in and to protect from progression to severe disease. And the targets here are much wider. This could include the spike, including the S2 domain, which doesn't usually is usually not the target of strongly neutralizing antibodies, but it's more cross-reactive, and potentially also the nucleoprotein, for example, as was discussed in previous talks. Now, just to contrast these two vaccine ideas, for a variant-proof SARS coronavirus 2 vaccine, we would like to cover, of course, wild-type SARS coronavirus 2, all current variants of concerns and variants of interest, and all future variants of concerns and variants of interest. And I think what would be very important here is that this also covers artificial escape mutants like the BMS20 that was published by Schmidt et al., and also mutants that come out of yeast libraries, for example, Jesse Bloomsburg, because this kind of gives us a glimpse of the future that could happen with these viruses in terms of antigenic drift. For a Pan-Sarbecovirus vaccine, all of this applies, but this should also cover all ACE2 binding Sarbecoviruses and all non-ACE2 binding Sarbecoviruses. And I think that's where we run into trouble a little bit. So there is, if you want to call it like that, some proof of concept out there, and this is actually data from a paper that Linfa Wang's group published, an excellent paper, where they also looked at people who had gotten the SARS coronavirus 1 vaccine back in the day and now were vaccinated with an mRNA vaccine against SARS coronavirus 2. And they looked at the breadth of immune responses, of neutralizing antibody responses that they got. And so the group that you should focus on here is this purple group. Those are the people who have seen both vaccines against SARS-CoV-1 and SARS-CoV-2. And what we see here is that they actually, after vaccination with SARS-CoV-2, developed a very broad neutralizing antibody response that, of course, covers SARS-CoV-1, but also other coronaviruses that are not members, or that are not SARS-CoV-2 or SARS-CoV-1, right? So we already see that there is the potential to induce these broad antibody responses. Now, there are limitations to that, and this is shown in this graph from another excellent study that looked at broadly neutralizing antibodies and their breadth. And what you can see here is a panel of antibodies that either bind to the receptor binding motif or to the RBD in general. They're all neutralizing, and you can see the breadth here, right? So we have a number of antibodies that have breadth, but it's not really crossing into SARS coronavirus 1, for example. If we look outside of the receptor binding motif, we see that antibodies can have broader coverage. But if we start to look into binding to spikes of viruses, Sarbecoviruses, that do not utilize ACE2, we basically lose most of these antibodies. And there is no reason to believe that that would be different with a vaccine. And so I think the challenge here is these non-ACE2 utilizing antibodies, viruses, of which we have a lot. Just a little bit about strategies and challenges here. I think there's some low-hanging fruit out there. We already know from this excellent work and other papers that exposure to SARS coronavirus 1 and then exposure to SARS coronavirus 2 already provides relatively broad immunity. And I think that's something that needs to be further explored. Exposure to wild-type SARS coronavirus 2 and then to Omicron may also induce broader immunity. There's not enough data yet to draw that conclusion, but I think this is something that really needs to be studied, and we really need to look at what epitopes are targeted here. In addition to that, there's indications that multivalent vaccines that contain several variants might actually provide broader protection, and that's something we have to keep in mind too. In general, I think vaccines that cover all ACE2 binding Sarbecoviruses have the highest chance of success. Again, broadening that to non-ACE2 binding Sarbecoviruses might be challenging. And even if we can get that, there might still be a problem with antigenic drift within SARS-CoV-2, because if a lot of people develop antibodies now to these conserved epitopes on the RBD, there might be antibody pressure and there might be antigenic drift. So we don't know how stable these epitopes are. And that's something to keep in mind too. And then, of course, there's the same issues that we have with the current vaccines. One is mucosal immunity, which is really probably crucial for sterilizing immunity. And the other point with the mucosal immunity is that secretory IgA has 4 binding sites. So it's usually of higher avidity as compared to IgG. And for influenza, it has been shown that secretory IgA with the same specificities has much broader binding and neutralizing activity as compared to IgG, which only has 2 binding sites. And then, of course, there's the issue of waning immunity, which is also an issue for current vaccines. But this will be the same problem for a more universal Sarbecovirus vaccine. Just a few thoughts about what is needed to move forward. I think we need a better understanding of cross-protection and protective mechanisms across Sarbecoviruses. Might not all be neutralization, but we need to really start to understand that better. We certainly need reagents and animal models to test cross-neutralization and cross-protection. There are models for SARS-CoV-1, but beyond that, there's not really much out there, and we need to generate these tools. We also need a better understanding of what type of cross-reactive immunity different exposures in humans induce. So, for example, SARS-CoV-2 vaccination followed by an Omicron breakthrough infection. This is work that has been done for influenza extensively. I'm showing data here on the right side, where cross-reactivity after an H1N1 infection has been assessed, and you see that basically the antibody response not only targets H1, but also targets other influenza virus subtypes. And we need to have a better understanding what happens for these coronaviruses in humans when different exposures happen. We need more research into non-ACE2 binding Sarbecoviruses to identify receptors, find suitable cell lines for neutralization assays and animal models. And I think we also need to understand what potential they have to cause outbreaks and to cause a pandemic in the future, right? We are focusing on ACE2 binding Sarbecos mostly, but there's many more that do not bind to ACE2. And I think the big question here really is, what would the regulatory pathway for such vaccines be? We've just heard that there is no pathway right now, and there's also no pathway for example, a universal influenza virus vaccine, right? It is not clear how these vaccines would be licensed, what claims you could have as a vaccine producer, and how you would really establish in humans that you get broad protection from Sarbecoviruses. And I know I had more questions than answers, but I'll leave it here, and I'm happy to answer any questions that you might have.
Well, thank you very much, Florian. It strikes me, and perhaps you would like to comment on this, that we need essentially all arms of the immune system. And specifically, would it not be valuable to induce T-cell responses that are broad? And I'm thinking here of the nucleoprotein, which has largely not been used in our vaccines. The inactivated vaccines essentially don't present the N protein in the way we would want it, but an mRNA vaccine or a live vaccine could stimulate responses to the nucleoprotein T-cell responses. And also, what about an attenuated strain that might give mucosal responses in the nasopharynx? I just would like your comments on those two thoughts.
Absolutely. I think if we want a vaccine that protects from severe disease, we should focus on T cells as well, and the nucleoprotein probably is a valuable target there. The only concern that I have right now is we know that these responses, T cell responses, are very cross-reactive within SARS coronavirus 2 variants. We do see a small drop-off. I think Alex Sette has shown that nicely for Omicron already. It's not concerning yet, but there is some drop-off. And I'm not aware of any data that really looks at that systematically across Sarbecoviruses, right? So we assume that there would be more cross-protection, more cross-reactivity on the T cell side. But I think the studies to actually show that with, a huge diversity of Sarbecoviruses are not really there. So it's an assumption, but I think there's research needed to actually look into that to see if that's really the case. In terms of mucosal vaccines, I completely agree. I think we need to put more effort into evaluating mucosal vaccines and moving them forward in the pipeline, not just for Pan-Sarbecovirus vaccine, but also for just the SARS coronavirus 2 vaccine. And something that would give mucosal immunity, I think, would be a huge advantage.
Mm-hmm. Okay, well, thank you very much for your thoughts on this. And I think now we should move to the panel discussion that will be moderated by Phil Krause. So, Phil, would you take over?
Yeah, hi, I'm back. So I'd like to ask the panelists to turn on their cameras. And that, I think, is Miles Davenport, Kevin Saunders, Eric Karakari Boateng, Helen Reese, Raymond Tellier, Farida Ismail Al-Hosani, Malik Perez, and Gustavo Mendes Lima Santos. And I think that, well, we have 8 panelists in about 40 minutes. And what we'd like to do is get a sense of what it would take to persuade you as panelists that a Pan-Sarbecovirus vaccine actually worked and should be used. So everybody talks about the regulatory aspects of this. And of course, one does have to persuade regulators, but even if a regulator were to authorize such a vaccine, there would be other SARS-CoV-2 vaccines out there, which perhaps would already have been proven. And so the question then is, what would be the level of evidence on such a vaccine that would persuade you as a scientist or an experienced person, not only that this should be made available, but it should actually be used and deployed? And so with that as a unifying theme, I'd like to ask perhaps one of the panelists to raise your hand and start off with an answer.
Yes, just a clarification. It's possible that Dr. Reese will not be able to connect because she is in a location with poor internet, just to let you know.
Okay, thank you. If we see her, we'll call on her then as quickly as we see her.
I'm here.
Oh, you are? Well, Helen, let's let you go first, just in case you lose your connection.
I shouldn't have said that. I should have kept quiet. If I drop, I apologize. I'm having a very unstable afternoon and changing venues. Well, I think the first thing I would say, and we've obviously got limited time, is there an unmet need? So do we need to be going down this route at all? And I would say that we do. But just listening to what we've said, I think at the moment everyone's talking about severe disease, which I think is right. But there was a reference right at the beginning to long COVID. And we haven't really thought about other, we obviously have talked historically a lot about transmission, but will we in the future have to be looking for new vaccines that are going to have some other characteristics in terms of protection? The second thing that really struck me, and Phil, thanks to you, is with a sort of partial regulatory hat on, is that I do think that that draft framework that you presented is extraordinarily useful. So I think that I'm not sure how the world will adopt it, but I think that we should be seriously looking at using this as a sort of backbone, but adding some of the questions that have come up from today's discussion. And the other thing that struck me listening to today's discussion is what a huge variety of approaches there are, looking for different outcomes and using different platforms. And given that regulators have got a vast variety of experience, I think it will be very useful to actually keep looking at these new approaches and saying, what is the regulatory pathway? And we heard from the developers also saying, what is a regulatory pathway? And then I agree with all of the comments that I think we must also be thinking as well about oral nasal administration. We mustn't forget the value of single dose. We heard from the Sonke, because in many resource poor settings, we are not managing to get more than one dose out anyway. So I think that we need to look at that. And the other thing that really struck me was this idea of heterologous. From the outset, you know, are we going to be looking for, with the challenge of a pan-coronavirus or new variants, are we looking already from the outset at primary series, from the outset, we're going to be looking at developers to develop that. So in terms of what we would look for, I think that this ongoing discussion about, you know, antibodies versus cell-mediated and, you know, severe disease versus an immediate response. I think that's going to be standard. We're going to be looking for all of that. But I want to reinforce something we've often also said is standardizing assays, because I think we've done some of this, but not nearly enough of it. So if you're saying what would we want to see in the future, it would be very helpful to have a standardized approach to evaluation, which is why I've referred to the framework. I think that's useful. Standardized assays so that we can compare like with like. A lot of the presenters are comparing their new products to EUL approved products. So I would want to see how they do perform. And lastly, the other thing I would want to see now for the world actually is how would they perform, and some of the data was shown, with people with pre-existing seropositivity. Because whether you're in a situation where you've got high vaccine induced responses, or in our situation in the African region where we've got high infection-induced responses, we might well be introducing new vaccines on the back of any of those. So I think that I would also want to say, you know, and that's where the one dose would come in, for example. You know, if you added something to people already exposed one way or another, what would a single dose look like? Thanks.
You know, you raise a very important point there, Helen, that people keep asking, but I don't know that there's a clear answer. And that is, as especially in the developed world, more and more people are being boosted with the original antigen. To what degree is that locking in some original antigenic sin, which might make some of these later approaches of presenting additional antigens less likely to work? I don't know if you have an opinion on that, Helen, or if others do, but this question of what to do against this background of seropositivity and previous immunity will certainly make the evaluation more complex.
I think it's something that would have to be looked at, but in terms of original antigenic sin, it would then, I think, depend on how different what's coming down the pipeline is going to look like and what the design of the new platform looks like. But it's certainly it's a thought, and I hadn't thought about that. Yeah.
Mm-hmm. Professor Paris, you put your hand up first, I think.
Yeah, Phil. So, I mean, in response to your initial questions as to what we would look for or we'd like to see in a Pan-Sarbecovirus vaccine, I think some of the talks by Ralph Barrick and others have shown the type of preclinical data that would be required. Clearly, if it comes to human phase 1 trials, looking at neutralizing antibody responses using a panel of surrogate neutralization tests like what Lympha has. He has a panel against a whole range of sarbecoviruses. So that would be the neutralization side of it. In terms of experimental animal model challenges, certainly you could use something like SARS-1 as a challenge. But I really want to focus on, I think, a plea that we heard from a number of manufacturers. I think the Janssen Group, the State Serum Institute, and also the other T-cell OCVAX group, about the non-neutralizing correlates of protection. And I must say, I am a neutralizing antibody person. A lot of my work over this pandemic has been on neutralizing antibodies. But recently, we saw in Hong Kong something that has made me sit up and take notice. So, as you know, we have two vaccines in Hong Kong, the BioNTech and CoronaVac. And at the level of neutralizing antibodies, BioNTech produces much better neutralizing antibodies against Omicron than CoronaVac. 2 doses of CoronaVac essentially gave nothing. 3 doses give something, but very low. But then my colleagues, Ben Cowling and my EPI colleagues, have just done the preliminary vaccine efficacy data against the huge BA2 Omicron outbreak that we had. And of course, BNT does well, even in the seniors, even in the elderly. But CoronaVac is not doing too badly at all. It is protecting from severe disease. So I think the non-neutralizing antibody part of it, and this could be T cells or the FC binding and other binding antibodies, I think if you're looking at protecting from severe disease, this is quite important. And then the other aspect of it is we have the breadth is one thing. The second thing is waning of immunity, which we see, regularly with the conventional vaccines. And of course, we know that with T-cell responses, certainly to SARS-1 17 years ago, those are still persisting. So I think we really need to pay more attention to how we quantify protection with these non-neutralizing responses. Over.
Thank you. So are you raising the question of whether we need Pan-Sarbecovirus vaccines in the first place if we think that the non-neutralizing mechanisms of protection from our existing vaccine might work and persist? Well, I mean, the example I gave you is talking about quite a diverse variant, which Omicron is. Now, as I think Florian just now mentioned, we really don't know much about how the T-cell responses will do against a lot of the sarbecoviruses, right? That has not been looked at. So that's something we have to look at as well. But I think, coming back to the question of the primary aim, protecting from severe disease, I think we really need to think hard about what we are trying to do and what the correlates are. Okay, thank you. Raymond Tellier.
Yes, thank you for a very interesting, indeed fascinating series of presentation. I must say I was very encouraged by some of the data already available, showing that you can broaden the virus protection with a Pan-Sarbecovirus or a pan-virus booster added to the current vaccine. This is not only encouraging, that might be very practical, and of course all the others approach are very encouraging. Now, I'm not an expert in vaccines. However, I wanted to share with you some concern that I noticed along several presentations, biosafety concern among some of the approach. There are people that have mentioned that we should have a broadly available large collection of coronaviruses that could be potentially emerging, so that they are available in the laboratory for neutralization studies, etc. There are a considerable danger of biocontainment and escape of virus from laboratory here, which is greater which would increase with the number of laboratories involved and not the size of the collection. And I am not here alluding to the origin of the COVID pandemic. I think that the consensus is very very strong that it proceeded like SARS, it jumped from animals. But we do have an important precedent, which is the flu pandemic of 1977, which almost certainly started from a laboratory strain somewhere. So these things are not impossible, and these viruses have a very well demonstrated pandemic potential. So we will have to be very careful about that. Also, I have been cringing since the beginning of the pandemic about experiment involving human challenge with SARS coronavirus 2 on volunteers. Even if they do not have severe acute illness, there is a considerable concern with long COVID, some manifestation of which seems to include nervous system damage, which may or may not resolve in time. So there are some serious ethical and biosafety issues there, which would be even more uncertain with other viruses about which we know even less. The other point, and this point I made in the chat, as far as I know, there are serious concerns to be had with the idea of a live attenuated vaccine for coronaviruses, because one of the properties of these viruses is that they create reassortant when a cell is infected with 2 different strains. Indeed, the joke is among coronavirologists that these viruses have a continuous segmented genome. And there is a well-known precedent for this in the veterinary world is that the infectious bronchitis virus of chicken, there are live attenuated vaccine for this viruses. And there's been several reports over the years that the vaccine strain recombined with a wild type strain and will give rise to an outbreak with the recombinant virus. Now, this is something that may be acceptable in the context of chicken farming, where you just want to increase the survival of a larger number of chickens so that they make it to the slaughterhouse, but I don't see that it would be an acceptable risk for a human vaccine. I noted concern about long COVID and vaccines, which is an interesting idea, because if it is in part based on an immune reaction or autoimmune reaction with cross-reactivity, you want to be careful about which antigen you inject. However, as the speaker noted, there has been to date no indication of that, and at the very least we can say that the risk of long COVID is much, much, much bigger with COVID than with the vaccine. And indeed, I've noted over the past 2 years, there has been some published anecdotal report of patients with long COVID that got better following a vaccination. The last thing I wanted to say as a clinical microbiologist is, whereas the potential of the N protein for T-cell based immunity should be explored, I would mourn the disappearance of one of the effects would be that the vaccinees would now very likely test positive on N protein serology. And this is a bit unfortunate, because right now with serology against the S protein and against the N protein, as they are getting better and better, we have a scheme to distinguish people who acquired immunity through vaccination alone or through infection and or vaccination. So that would be too bad that we lose that, but of course that's not a major obstacle. Finally, I was very interested in the comment of Dr. Perez about and many other presenters about the possible action of non-neutralizing antibody. One thing that has been striking me since the beginning of the pandemic is that there might be benefit for antibodies that trigger the complement, because we are dealing with an envelope viruses. So these are all the thoughts that I have. Thank you.
Great, thank you very much. So Dr. Saunders, you don't have your hand up, but I'll turn to you next. You gave really, I thought, a masterful description of different approaches that could be used to address this problem, I guess all of which you're working on now, but which means that you probably have given some thought about how you would decide which of these approaches work. And especially as you're thinking about multiple different approaches, at some point you might want to narrow these down and decide which ones you think work better than others. And so what are your thoughts about how to evaluate these vaccines, either in the short term or ultimately in the longer term to decide whether they should be used?
Thank you for the question. So we have thought about this, which is one of the reasons why we broadened beyond just neutralizing antibodies. And so what we're looking for is a vaccine that can combine targeting these conserved epitopes and then also being able to bring in T-cell responses that might be a little bit broader and can contribute. We don't think that just by itself the T-cell response will be sufficient. We do still think that you need to have neutralizing antibodies that can recognize multiple different coronaviruses. So the way that we've approached this is really kind of with two different end goals. So one is to have a vaccine that works against most of the beta coronaviruses. And what we've done is we've taken the different S protein sequences and we align those sequences, and we can quickly see how S proteins differ. When we do that, we can find that within certain groups like 2A or 2B or 2C or 2D, you can find a relatively high homology among those S proteins. So our approach right now is to have vaccines that can react with a couple or a few or some number of S proteins that are in each of those groups. We feel pretty confident based on the conservation of sequence that if you can have an antibody that recognizes, 2 or 3 of those different S proteins in that group, that we then would have some reactivity against a virus that may come along in the future that then falls within one of those groups. And so that's the strategy, so to speak, that we're using for development of a beta coronavirus vaccine. When you start to move beyond that, it becomes more complex. So if you're talking about alpha coronaviruses, I think there was another talk earlier this morning that talked about delta coronaviruses as well. And so if you're talking about those types of vaccines as well, then it becomes a little bit more complicated as to how you can bring all of those different antigens into one vaccine. And so our approach for that is to do a very similar approach, but we're essentially looking at, can we target conserved sites within alpha coronaviruses? Can we target conserved sites within beta coronaviruses? And then we basically can bring those two vaccines together. I think it's a fairly difficult problem. I think it's not as straightforward as we might make it seem, even with some of the presentations that we showed today, where we are starting to extend the breadth beyond just SARS-CoV-2. It still really is a difficult problem. And so we want to combine not just our current mosaic approaches, but I think there's also some engineering that has to go into the thought of what you display to the immune system so that you get the optimal response with as few antigens as possible.
Sorry about that. Do you think there might be a price to pay in trying to gain breadth in terms of efficacy against any individual virus? Where, so one idea is that these new vaccines could be evaluated as though they were SARS-CoV-2 vaccines and compare them with existing SARS-CoV-2 vaccines. And if they work well, then one can start using them. And then if they also have these other advantages, then we'll get them into use and there would be an argument in favor of using them. But on the other hand, if they might not work as well because you've traded specificity for breadth, then maybe that strategy won't work. Do you have any thoughts on that?
So I think our approach is to do both. So that means that we want to target a site that's conserved on SARS-CoV-2 as well as SARS-CoV-1, and then moving on to bat coronaviruses that might be related. And if we target that site and generate a highly potent response against that site, and we think that's the advantage of the nanoparticles that most of the folks talked about earlier, is that you can really increase the titers and you can really get a high level of antibody response. And if you can do that, then you target these sites that are not only on the bat viruses that are circulated in those current species, but also on SARS-CoV-2. And in doing so, you actually get activity that's helpful for the current pandemic as well as future pandemics.
Fantastic, thanks. Dr. Karikari Boateng, would you like to comment on this question of evaluating new SARS-CoV-2 vaccines? Or actually, sorry, new Pan-Sarbecovirus vaccines. Apologies. Oh, I can't hear you. I think you're still muted. Still not. You might, if you're using a computer, you might be able to unmute with your space bar.
Mena, can you please unmute him? Maybe that will help.
Oh, it looks like he disconnected and is reconnecting. So let's turn to Dr. Davenport and ask for his comments. Miles, you've been a strong advocate of neutralizing antibodies as correlates and what we can learn from those. So be interested in your thoughts. And you're also muted, I think.
Yes.
Can you hear me, Phil, now? Hi,
Oh, you're
Phil,
back.
can you hear me?
Actually, you know what? Since we have Dr. Kariboteng, let me save your comment, Miles, and let's have him come back. Yes, we do hear you now.
Okay, sorry. I think I had an interruption with my connection. Yeah, in addition to what the other people have said, I quite remember when all the presenters, especially developers, were talking, what they said was, what is a regulatory pathway? What is a regulatory pathway for such vaccines in case, like when they get to the clinical stage? The only thing I have to add to what the others have said is, we would have to look at that immunogenicity. When we might get to we have to look at immunogenicity and then bridge it to non-human primate studies. And in addition, add real-world data as was done during just recently for SARS-CoV-2 vaccines. Yeah, that's what I want to add. I can't hear you, Phil.
Sorry, my fault now. Thank you very much. And let me just turn to Dr. Davenport now.
As you suggested, I'm a little bit of an advocate of neutralizing antibodies, and I'm a little bit surprised to hear the amount of discussion about T cells, because I understand they're very attractive because of the high level of cross-reactivity between strains. But to me, that's also the evidence that they're not actually providing protection, because when we get a new variant, although it's recognized almost as well by T cells, we see a loss of protection from both symptomatic and severe disease. So the evidence that has been discussed earlier was that T cells might provide protection in the face of waning immunity, but we see protection wane exactly as predicted by neutralizing antibodies, but also T cells wane at the same rate as antibodies. So you can't really differentiate there. But the real difference is when you do come to the variants, that you see a significant loss of protection from both symptomatic and severe, while maintaining fairly constant T cell recognition. And the most recent case being between the Delta and Omicron, where the studies show no significant difference in loss of T cell recognition compared to the vaccines. And yet the efficacy against progression from symptomatic to severe disease is much higher. The progression from symptomatic to severe disease is much higher in Omicron, consistent with the loss of neutralizing antibody binding, but not consistent with the maintenance of T cell recognition. So I think there's a lot of work to be done to establish in what circumstances T cells are protective and to find them, to define the correlation of protection once you correct for the obviously potent effect of the neutralizing antibodies.
No doubt that's complicated. I will comment, though, that I think this literature is a little bit more complex than you're making it sound, including that, for instance, with the people who are going to the hospital with Delta versus Omicron are probably very different people. And for people who are vaccinated, there's a smaller proportion of people are hospital. And so it may be that the apparent reduced efficacy of vaccines against severe disease caused by Omicron may also be related to the fact that the people who are going to the hospital with Omicron are fundamentally sicker people who have more risk factors, and the vaccines wouldn't have worked as well if you were able to identify those people also with Delta. So I'm not completely persuaded that there is a dramatic drop-off in protection against severe disease with new variants. And that's obviously could be the topic of a whole other meeting, but I'll just say that might be a little bit controversial. Okay, well, still have a few minutes left. Are there Others on the panel, do any of you on the panel want to make any additional comments, in particular on how you think a Pan-Sarbecovirus vaccine gains traction in a world where there will already be SARS-CoV-2 vaccines and many people will have been vaccinated with it. And obviously, if these development efforts are to be successful, there will need to be a market for it. And so there will have to be data that suggests that this is something that is really worth taking. And so what does that data look like? Dr. Saunders.
So since I didn't see anyone else jump at it, so I think I'll just try. So I think what's going to be the distinguishing factor from the current vaccines is whether or not the vaccine is effective against the non-ACE2 using coronaviruses. So when we study mRNA vaccination, we find that there are cross-neutralizing antibodies generated in humans, generated in monkeys, generated in mice, and they can neutralize many of the SARS-related viruses. And so I don't think that that's really the target here. I think what we're really looking for is, do you expand the breadth to Sarbecoviruses? Can you protect against some of these other strains that are not going to use ACE2 and are not going to fall within that SARS-related virus category? And if you start to see protection against that, I think it's going to distinguish itself from the current mRNA vaccines that's out there.
I guess my comment would be that I'd love to see a pan-strain vaccine for SARS-CoV-2, and that until we have something which can achieve that, I think it's a big ask to look at a Pan-Sarbecovirus vaccine.
That's true, although it sounds like many of the strategies that have been discussed today could be used towards either end. And I take your point and Florian's point as well, that trying to go broader will be more challenging, for sure. And so it may be that biting off something a little bit smaller is a simpler first step. Helen, a final comment from you.
No, I mean, in a different angle, but I mean, you plan for the worst and hope for the best. So I think from a scientific point of view, we need to proceed. I think a different question will be the enthusiasm of the public to keep taking vaccines. And the thing that would change that is if there was a new variant, for example, that suddenly threw us back into a sort of Delta type scenario again with high hospitalization rates. That will get people vaccinating again. The problem, as we all know with Omicron, is that there's a sort of lull in enthusiasm. So there is a public problem here, but I think from a scientific point of view, we need to be ready and we should be driving this because we should anticipate, as we all know, that this, in some form or another, we're going to be stuck with SARS-CoV-2 for a while to come. Thanks.
Okay, thanks. Well, I think there's general agreement that this is an absolutely critical public health effort, because if we don't find a way to address a broader array of sarbecoviruses, to anticipate variants before they occur and protect people, then based on what we've seen so far from a public health perspective, certainly the risk of something bad happening is high. And of course, as Helen says, if nothing bad happens, that will be very good. But at the same time, given the opportunity and given the very exciting science that we've seen today, it seems like we really do need to proceed. So I will close this panel with that remark. And I think there's one final panel, which one final set of speakers that Stanley will introduce.
Yes, well, thank you, Phil and the others. And our next speaker is Christina Cassetti from NIAID. Christina?
Can you hear me?
Yes.
Okay, wonderful. Thank you very much. And thank you all for the opportunity to tell you a little bit what my institute is doing on pan-corona vaccines and some of the new and exciting programs in this area. So NIAID realized quite early on in the pandemic that since SARS-CoV-2 was the third clinically important coronavirus that emerged in the last 20 years, since coronavirus has been evolving constantly throughout the pandemic, as we heard, it was very important to start developing pan-coronavirus vaccines. So in November of 2020, which was 8 months after the WHO declared a pandemic, we issued a funding opportunity to support the development of pan-corona vaccines, which of course includes Pan-Sarbecovirus vaccines, but also Pan-SARS-2 variant vaccines. And during the last year, we made 4 large awards to 4 academic institutions, and some of which you heard before during this meeting. So the awards were made to Duke University, University of Wisconsin, Brigham and Women's Hospital in Boston, and Rockefeller University. And we expect to make more awards later this year, obviously pending availability of funding. But the overall goal of these are large PO1 projects are really threefold. The goal is, number one, to better understand the exact human responses to SARS-CoV-2 infection and vaccination that have an impact on efficacy and breadth of the immune responses as a way to really better inform the vaccine design. The second goal of these projects is really to use structural guided molecular and computational approaches to really design the best immunogens that elicit a broadly neutralizing antibody response and also T-cell response. And they're looking at different parts of course of the spike protein and other antigens as well. And the second part is, of course, to exploit a variety of platforms to deliver these antigens, including VLPs, alpha virus replicons, particles, protein nanoparticles, mRNA-containing lipid nanoparticles, mosaic nanoparticles, and also there are some efforts to develop adeno-associated virus vector platforms for an intranasal delivery of vaccine. All of these with the goal of increasing the breadth of protection of vaccines and hopefully also longevity. And in addition to these extramural awards, of course, we have a very active intramural group, different intramural groups working also on these broadly protective vaccines. One of them, of course, is the Vaccine Research Center, as well as our intramural laboratories. And they're also pursuing different approaches, mostly leveraging our efforts on universal influenza vaccines, including nanoparticle-based vaccines, where they take the whole stabilized spike protein of multiple different coronaviruses. This is for the VRC, or also an approach using a variety of inactivated viruses, like they're doing for influenza as well. So this is like high level picture of what our institute is doing. And I'd like to end by mentioning very briefly a very exciting collaboration on pan-corona vaccines that has been developed between our institute and CEPI. So since our institutions are probably some of the largest funders on pan-coronavirus vaccines, so we have decided of establishing a common scientific steering committee to accelerate the development of these vaccines, help guide the investigators with some of the best and brightest minds, and really push for development and clinical evaluation and testing of these vaccines so that we can be better prepared for the next variants that they're likely to come our way. And that's all I have.
Thank you very much, Christina, and that illustrates the importance of NIAID, but also, if I may say, because of my association with CEPI, it shows the value of an organization that is looking ahead rather than trying to only cope with the current circumstances. Well, again, thank you. And now, according to the program, WHO may have some comments. Ana Maria, is that the case?
And thank you for the introduction about organism. Can you hear me?
Yes.
Ana Maria, were there two other speakers in this session, or did they are they all?
This is what I was going to ask.
We have Dr. Yves Levy and Dr. Chris da Costa as well.
Yes. I was going to say that there are other organizations thinking ahead of the game, Stanley. So we should invite Dr. Yves Levy and Dr. Chris da Costa to speak. If you
okay, fine. I didn't see them on the written program, but please, let's see, who is.
Yves, do you want to
Yes, thank you. Yves Levy, so perhaps I can start just to mention some points around the Vaccine Research Institute from INSERM, what we have launched in the last 2 years. And so I have discussed and presented, and thanks to the WHO, the strategy for the vaccine, and this followed the previous session, because as you understood, we are developing a protein-based vaccine and also including exactly some of the points discussed today with T and B cell epitopes. And I think we need to address, and this is the goal of the VRI, the Vaccine Research Institute, first, how to induce long-standing immune responses, because I think we will face a fatigue of the population to receive several boosts within a year and to try to run after the different variants. The second point is to extend the protection and to prepare perhaps the next pandemic, and this was discussed. And the strategy we have developed is somehow very close to what Dr. Sanders mentioned. We tried to align and we worked on the approach to identify the T and B cell epitope that will be contained in a vaccine, and taking advantage of the last years of development of this dendritic cell targeting vaccine, which has several advantages as an immune stimulant and also to improve the delivery of the antigen. And so the goal is to develop this Pan-Sarbecovirus and to be exactly in the situation where we are. So to have a booster vaccine, an efficient booster vaccine to extend the response. The other point is to try also to understand the physiopathology of the long COVID, because we need to protect perhaps from the infection, from severe disease, but also for the long-term complication of the infection. So this is one of the aspects. The other point we have developed in the Vaccine Research Institute is to try to understand the physiopathology, and this linked with the other initiative from INSERM. There is a large French COVID French cohort of patients with the COVID, and after the first waves and the second wave and the other waves. So we try to understand the physiopathology of this. And the third point is within some European funded programs to study the prevalence of the infection and to study the durability of the natural immunity with the colleagues from African countries. And we have also developed a European program based on neutralizing antibodies, and so new neutralizing antibodies, which will be in clinic in the next year. The other point is that the government, after the lessons from the COVID crisis, launched a very important program in France, and INSERM is part of this program, which is a program funded by 7 billion euros to try to organize and to develop new strategies in research, including emerging disease and vaccine, and also to make the link with CDMO manufacturers of the vaccine to improve the bioproduction capacities in France. And the last point is the success of these new programs is based also on the new organization of the research and development of vaccine in France. And so two big organizations, INSERM and the CEA, based on the Vaccine Research Institute in IDMIT, so two programs in France, they joined their effort to try to develop a new institute covering from basic science to preclinical, clinical toxicology, regulatory affairs, and bioproduction with the link of the industry partners to have a new pipeline, a new institute that cover and which may develop new vaccine with unmet vaccine needs today, and vaccine that may help France to face the new pandemic. So INSERM is part of that, and the Vaccine Research Institute will be the ground with the IDMIT from the CA of this new institute. So thank you.
Well, thank you, Yves. And as a Francophile, I am delighted to know that a new institute is being developed to maximize French science. So that's great.
Thank you very much.
Sorry, I didn't catch the name of the second speaker.
Dr. Chris Da Costa.
Okay, can we? Yes, Dr. Da Costa.
Yes, thank you very much, Stanley, and thank you for inviting me to talk about CEPI's program to support the development of broadly protective beta coronavirus vaccines. We like to use the term broadly protective for some of the reasons that have been very well articulated during this session as to what exactly is meant by a pan-coronavirus vaccine or a Pan-Sarbecovirus vaccine. Next slide, please. So at CEPI, we do a lot of our work through calls for proposals where we fund various developers, prospective partners to develop these vaccines. And since about a year ago, we opened up a call for proposals for both what we call broadly protective beta coronavirus vaccines, but also those that are more targeted to SARS-CoV-2 variants, the broadly protective SARS-CoV-2 variant targeted vaccines. And the premise for this call was based on innovative immunogen design that was targeted through a number of approaches and a number of principles. First of all, to develop platform technologies that could deliver vaccine immunogens in a manner that would elicit broad protection, but that would also be very highly adaptable and capable of rapid response in the event of emergence of a novel variant or a novel coronavirus, so that the rapid response could then be brought into play. The approach has been targeted on various strategies, many of which have been discussed earlier today, one of which is the multivalency approach, using several different spike proteins, RBDs, or other immunogens attached to some sort of a backbone or platform. But also very heavily weighted on intelligent computational design using state-of-the-art approaches to looking at highly conserved epitopes, consensus sequences that could portend to broad protection. We've also had approaches based on monoclonal antibodies to select broadly conserved or cryptic epitopes, not only within the Sarbecovirus genus, subgenus, but also across other subgenera within the beta coronavirus genus. And so we work very closely with a number of centers of excellence. Several of you are in this meeting that we work with. Some of you have been funded. Others of you are in the process of waiting outcome of the applications. And we really look for structural biology approaches that would predict the design of relevant immunogens to achieve the goal of broad protection, breadth of protection. The next slide, please. This schematic just really shows the bookends. There's discussion of what really is a Pan-Sarbecovirus vaccine. You have on the left-hand side our focus on the SARS-CoV-2 variants of concern that are either current variants of concern or pre-emergent variants of concern that we presume might continue to evolve. Through a whole spectrum, through those candidates that have a combination of Sarbeco and Merbecoviruses, which we see a lot of. Of course, MERS, as was mentioned previously, is really the only representative in those vaccine designs. And then across the whole spectrum, we do have those candidates that try to broaden breadth of protection, achieve that through multiplicity, multivalent, multiplex immunogens from various SARS, MERS, and potentially also in future other subgenera of beta coronaviruses. We remember the PREDICT program from 2009, in which USAID looked at the spillover potential for coronaviruses in humans. This revealed well over 100 coronaviruses with at least theoretical spillover potential. So We also theoretically try to target as broadly as possible. We heard an earlier speaker talk about the complexity of that approach. These all have timelines which vary depending on how broad one wants. Of course, for the SARS-CoV-2 variants, we want to achieve very rapid clinical proof of concept within a couple of years, whereas for the others, more broadly protective, we expect that they're going to take much longer to come to fruition. The next slide, please. This slide really delves into what we actually have in our portfolio at CEPI, and this is the CEPI-funded portfolio, both for the first generation vaccines that we all know very well now, some of which have received emergency use authorization or licensure from various regulatory authorities that CEPI has had a hand in, including Moderna and Novavax and BioE and AstraZeneca Oxford, as well as the second tier vaccines, some of which also you're very familiar with, like Gritstone, SK Bioscience, Walvax, etc., that are currently supported by CEPI for further development. We also, more pertinent to this talk is the blue row there with SARS-CoV-2 variant targeted vaccines, of which we have fully funded BioNet Asia for an mRNA approach. We also have others that are seed funded with smaller amounts of funding to achieve preclinical proof of concept, with a view to further full substantive funding from CEPI should they achieve a candidate that demonstrates preclinical proof of concept. And these include Vido in Canada, Migvax in Israel, and Affinivax here in the USA. Finally, we also have the more broadly protective approach, more long-term approach. And we recently funded Pan-Sarbecovirus in India, Diacinvax in the United Kingdom, and SK Bio Korea has also been previously funded. This is a range of technologies, as you can see, from protein-based to DNA, to RNA vaccines, some viral vectored vaccines in the earlier portfolios, and we're looking to see if there may be some coming forward for the active portfolio for variant vaccines and BPVC as well. There are others, if I were to present this in 2 weeks or a month from now, it's going to look very differently because we're on the brink of funding a substantial number of additional candidates, both for variant targeted vaccines and for seed funding, as well as for full funding for broadly protected vaccines. And the next slide, please. And so just the last couple of slides go into a little bit more detail with our various funded projects. This is for the what some would consider Sarbecovirus restricted variant targeted vaccines. BioNet Asia, with substantive full funding, is based in Thailand and has an mRNA LNP approach with a multivalent immunogen design approach, intramuscular route. Migvax in Israel has a protein similar to Vido as well. These are protein-based. Also, one is intramuscular, one oral, also based on multivalent immunogen design or intelligent design. Affinivax in the US is a protein with a polysaccharide backbone that also has multivalent immunogen design. And these protein vaccines are all adjuvanted one way or another. The next and the final slide really goes into some of the broadly protective, as I mentioned before, that have been fully funded. And we have a number of different countries. Again, from India, we have Panacea Biotech, from the UK, Diacinvax, and South Korea, as I mentioned previously, nanoparticle-based vaccines for Panacea and SK, and mRNA with LNP for Diacinvax. And these are all very strong on immunogen design that use a variety of approaches. They are largely two-dose regimens, largely intramuscular, although we do have an intranasal approach possibility with Diacinvax. That's, I think, concludes the presentation, just a brief snapshot of what we're doing at CEPI. Thank you very much.
Well, fortunately, that is quite impressive, and very happy to see that. Phil and Ana Maria, I have a few very short final comments, but would you, either of you, like to
Yes,
make some
Stanley.
comments?
Yes, Stanley, I have some slides to show I am in your good books of people who looks to the future. And you have to say amazing presentation when I finish. Okay? So
Yeah.
next slide, please, Jimena. So a little bit of history. So we in WHO are working on this area on Pan-Sarbecovirus and on epidemic diseases because our 194 member states decided we should do so in 2015 after the Ebola epidemic in West Africa. And this is a global strategy and a preparedness plan that was developed to be activated, and it has been the place during COVID pandemic. Next one, please. So why do we do this beyond our 194 member states? That was not enough. In our recent consultation on global research and innovation, Professor Joseph Stiglitz reminded us that the benefits, the economic benefits of investing on and developing new vaccines and new interventions is more than we are losing economically for the impact of the pandemic. So as Helen and Phil were saying, definitely we need to continue to work on this area as soon as possible and move forward to not have any regrets in the future. Next one. We not only work on COVID, we also work in other priority pathogens. And this is just a graph to show that we have a systematic approach that perhaps we need to pursue for the Pan-Sarbecovirus vaccines. This consultation is the first step of developing a roadmap. We need to define, as several people mentioned here, a target product profile. We need to discuss what are the attributes that from the public health perspective these vaccines should have, and there is some debate about what kind of vaccines we should develop, and certainly a discussion on the TPP will help that. We need to discuss how we are going to evaluate them in terms of trial designs, to have some trial simulators and innovative analysis as we do for other vaccines. And part of this is the framework that Dr. Krause shares with you. Next one, please. I want to cite Stanley, who during the consultation mentioned that aside from welfare, the greatest threat to human race is pandemic disease. And because we have made tremendous progress in the last 2 years, and we have done a lot better since polio, we should continue to explore multiple strategies for developing vaccines. This is part of the consultation today, part of our global commitment to move forward. Next one, please, Jimena. We just published our research priorities for COVID-19. It's in our website. And I just want to underline that Pan-Sarbecovirus vaccines is one of the global research priorities from the perspective of WHO and our member states. Next one, please. So how can we help? We want to help and not be on the way. So if there is any way that we can help, so we are proposing, and this is for debate, that, and you know, we have very good relationships, working relationships with INSERM, with NIH and NIAID, and with CEPI, but there are other developers who are not part of this process. There are other developers who maybe are trying in isolation, and we heard from some of them today. So perhaps if we have a forum where we can share the data, the progress, and maintain each other updated on the newest strategies, it's an option that we have used in the past for other vaccines and have produced good results. In addition, we think that by sharing, we are going to perhaps have economies in terms of reduction, refinement of what we do, and replacement that could be addressed. And so next one, please. So what we are proposing, and this is for debate and for discussion, is to maintain regular interactions where we review the process. We also want to identify the scientific bottlenecks and have dedicated discussions. Today, for example, people mentioned that if the regulatory bottlenecks should be addressed. Others talk about sharing of materials. There were discussions about how to design the studies, discussions on the selections of the components of this vaccine and the breadth of antigenic content of the different vaccines. So should we discuss this together rather than having parallel efforts, perhaps not making the best use of global public health? So WHO's aim is to contribute to accelerate the candidate vaccine assessment and pathway. And we want to work, and we are very pleased that very renowned people like Stanley has agreed that this is important and has agreed to help us push this agenda forward because we think it's important. Next one. So I know that there are individual initiatives, we know that, but we want to discuss how can we work together, how we can potentiate the efforts of these different initiatives. And moreover, how can we discuss whether or not these vaccines that we are developing are the response to the public health needs. And I think this is where we at WHO have a comparative advantage. So I want to invite you, the panel members and the speakers, for a short period of time to raise any questions, any points that we should consider for our next deliberations, any issues regarding coordination or any support needed. And I invite you to please do not repeat things that have been already highlighted by other speakers because we already took note of that. Thank you, Stanley.
Well, thank you, Ana Maria, and I appreciate the WHO's important contributions in the future as well as currently. Phil, would you like to make some closing remarks?
I could, but I think Ana Maria is hoping that some of the panelists will make put on the table at least a few ideas for topics that could be addressed in this forum in future meetings. And to kick that off, I will say I think we had a good discussion about what some of the advantages of more broadly protective vaccines will be. I think that the manufacturers, to the extent that they participated, did express some concern about what the regulatory and use pathway for this will be. And so I suspect that we could productively have another meeting at some point in the future to try to delve more deeply into that, involving more regulators and perhaps more data and more concrete proposals about how it is that these vaccines could actually be evaluated and brought into common use.
Yes, thank you, Phil. Maybe I could use your style and invite Christina. Christina, do you want to help us discuss if, from the perspective of NIAID and your initiative, what else should we do to pull this global energy and advance the development of these vaccines?
Thank you, Ana Maria. Right now, at least I'm talking from our perspective, or US government funding, we are a little concerned about sustainability of funding to continue all these projects. It seems that, I guess, the perception is that as soon as the pandemic ease up a little bit, the people want to move on immediately to the next thing. And we think it's super important for us to keep the momentum of all these projects from obviously from a funding standpoint. And this is very much in our forefront of our thoughts at the moment. I mean, there is a lot of interest from the scientific community, but there has to be sustainable funding from large funders to complete these projects and bring these vaccines, for example, that we discussed this morning, all the way at least to clinical evaluation and some sort of approval, so that we are ready for the next wave to come.
Absolutely, Christina. Thank you very much. Mary Paul, are you still there? Do you want to add a few points? Mary Paul? I think she's multitasking. So okay, I just want to add a couple of more people, Stanley, to talk before we close, because from WHO we would like to hear really, like Christina did and Phil, what do they think are the critical elements that we can help to push forward in this topic. So maybe I can ask Dr. Barry, you are there. can you hear me? And can you please tell me from your perspective, what should we do next? What is the next topic that we should highlight as important in this area?
Well, thank you. Yeah, that's a good question. And I certainly think we have a variety of platforms that are in play that are fortunately well supported. And so those leaders at this moment, I think, will provide very valuable information and direction for discriminating between and prioritization between the different vaccine platforms. I think there's a variety of individuals in this discussion group have pointed out there definitely need to be additional campaigns for reagent, virologic and immunologic reagent development as we move from the more simple vaccine platforms for, let's say, Sarbecoviruses to the more complex platforms of Pan-Coronavirus-based platforms. And a stepwise process in that, seems to be most prudent because it is low-hanging fruit to stay more focused. And as we move from one platform to the other, we will be informed about potential problems and issues that need to be considered earlier in the process. And so I think that's important as well. Probably needs to be some resolution about the importance of spike neutralization-based platforms and the role of T-cell immunity in control and regulation of disease. Personally, I'm a fan of a combinatorial-based strategy. I'm personally not convinced that a simple T-cell based vaccine platform will protect from either serious life-threatening disease or lethal disease. It may reduce lethal disease, but I don't think it will be of significant impact to build wide support upon public use. And so those are sort of my takeaways from this conference.
Thank you.
I hope I answered your question.
Yes, you answered a question and you raise again an area of debate. Dr. Da Costa, do you want to add? I see your hand is raised.
Yes, thank you very much, Anna. I just wanted to say a few words to complement what Dr. Cristina Cassetti said, about the partnership. We are working together with NIAID at CEPI to forge a partnership we're very excited about. I think it's very important that we complement each other's portfolio and we are seen to develop products that are complementary. Christine talked about the limited funding through NIAID. CEPI has probably got deeper pockets because of our various stakeholders, and there's a lot we can do to move products forward that do achieve preclinical proof of concept. So I think that's a very important example of how things should be done moving forwards, is working in partnership. We hope other organizations also, join us in this partnership in future, and I think this is going to be a very big part of what we do. The final thing I wanted to say very quickly was about the regulatory pathway forward. I think that's going to be very key to these novel approaches and these novel vaccines to define exactly how you demonstrate that breadth of protection from a regulatory standpoint is really going to hold water. It's going to be, I think, a challenging concept. And I think that's something that needs a lot of attention to be paid to it. Others have spoken about the breadth of protection. What exactly do we mean by broad protection, and how do we demonstrate that? Do we look at a panel of various viruses across subgenera of beta coronaviruses that would be representative? Do we look at their receptor binding and make sure that we cover all strains? It seems it will be challenging, but it's going to be an important thing to do going forward for these broadly protective vaccines. Thanks very much. Just a few things I wanted to mention there. Yeah.
I want to invite now Dr. Malik Paris to give us his views. And Yves Levy, can you please prepare, because I will come to you after, Dr. Paris. Malik, are you there?
So I wasn't clear. What was the question you were posing?
The question is, how can we continue to work together? What are the next topics or the next actions that we should take at global level, and how can WHO help facilitate those actions?
Well, I think there's a lot of unfinished business from where we are at the moment. We the topics we were discussing today, including the different breadths of protection. I think Florian summarized that quite well in his talk. We are talking about Pan-Sarbecovirus or pan-coronavirus protection, but we do have, first and foremost, the challenge of protecting against the next variants that come our way. So I think that is a top priority. And then we can move to the sarbecoviruses and to other things. And I'm not completely sure whether these different targets need somewhat different approaches. So I would be a bit cautious about purely focusing on sarbecovirus cross protection versus trying to be protective against the SARS coronavirus 2 variants that might come our way, which is the imminent problem. Over.
Thank you very much. Yves, are you there?
Yes, thank you.
Can you help me with this question, please?
So two points, next topic, next action, and the strategic view. Next topic, I fully follow what Phil proposed. I think we need a meeting to discuss a regulatory process. We see that there are different other platforms under development. So I think it's key to have a global scientific and regulatory agencies discussion all together to see what could be the endpoint and what could be the next step. This is key for biotech, for pharmas, and also for the scientists to see exactly what are the next trial to do and what are the paths for regulatory and to extend the portfolio. The second point is I would love to see also an extension of the CEPI collaboration. I think we mentioned NIAID and CEPI. I think for the Vaccine Research Institute from INSERM, I think we will be very also interested, and also for the new institute to collaborate with CEPI. I think that regarding the global actions, ACT-R or COVAX or the place of CEPI, and I think it would be very interesting to see if CEPI could also extend this collaboration to other institutions or research organisms. And I hope that the WHO could help to organize this or to follow this and to help the different institutions to build this new collaboration that we need for the future. Because the problem of sustained resources will be a very important problem. And France, as other countries, they have the same, but the level of the funding is not exactly at the level we need to develop these new platforms and to address the next challenges.
Thank you, Yves. Dr. Walid Abdurham, apologies for pronouncing your name badly. Dr. Walid, the floor is yours. Dr. Walid Abdurham. Maybe I am mispronouncing the name terribly. Stanley, can you help me?
It was Ali Jabbar.
Ali Al-Jabbar? Okay, while we resolve that problem, I will try Mary Paul. Mary Paul, are you still there? And Dr. Mary Paul? So let's hear from Africa. Dr. Karikari Boateng, do you want to give us your perspective on how WHO can help with this and what are, in your view, the next steps?
Yes, Dr. Anna Maria, there's one thing I want to add. In case these vaccines they proceed to the clinical development phase. I mean, this disease hits all continents. So when the clinical trials are being planned and all continents, there should be equitable distribution of the trials among all continents. It should not be like what happened in COVID, wherever just only a few, of the trials took place in Africa. I can just mention it only took place in some few study subjects in South Africa. I mean, that thing should not be repeated. I mean, there should be equitable distribution of the clinical trials across all continents. Over.
Okay. And I just didn't see, is Dr. Gustavo Mendez Lima Santos connected? No. Okay. I will try the last time. Dr. Walid, do you want to speak? Can you unmute yourself? Otherwise, we will move on, Stanley. I think we failed. So Stanley, I give back to you, or I give back the floor to Phil?
Well, so let me conclude the meeting. I won't take very long because I think, whoop, yes?
Phil has a summary, Stanley.
Oh, I was not aware of that. Phil?
So, so what I did is I was listening, I just produced a few slides to summarize what I heard, but I doubt it's so different from what you heard, Stanley. Rather than go through the slides, perhaps I can just say that what I heard was still a very strong argument in favor of more broadly protective vaccines. And there were a lot of very useful suggestions about what could help. And this includes challenges that were raised in terms of understanding the breadth of responses, the breadth of viruses that can infect humans, how it is that these viruses are drifting, identification of conserved epitopes and assay development, including having panels, more work on models, small and large animals, more work on COVID-related ARDS, vaccine-enhanced respiratory disease, or possible enhanced, right? And a better understanding of what can be done with human challenge studies, as well as the role of pre-immune status, kinetics of antibody response, of immune response, and correlates of protection. There is, I think, it was an interesting discussion. It seemed like most of the developers felt that it was important to come up with very strong cell-mediated responses in addition to human responses. And I think a better understanding of exactly what it is that is protecting and likely to be protective for these vaccines is going to be very important as well. But it was gratifying that what the manufacturers said was very much the same as what I heard the scientists say. What the manufacturer wanted to hear more about is what's the target product profile for these vaccines? What do we want them as a scientific community to achieve? What are the considerations for testing them in seropositive populations? And again, how to standardize T-cell assays. WHO, I think very importantly, is offering a forum for follow-up discussions to facilitate the exchange of ideas and to help with research coordination. And I think we heard that as funding and global interest is in the pandemic maybe waning just out of fatigue, I think maintaining a high degree of interest in this very important topic is going to be one of the critical challenges going forward. And so I strongly recommend that those of you in the community take advantage of this offer from WHO, and in fact, think about how to plan meetings that will make a difference and help keep up the momentum as well as facilitate the exchange of ideas. And then the other thing is just a thought, which is that what we learn about increasing the breadth of protection against SARS-CoV-2 will inevitably have additional future implications, because these kinds of science don't occur in a vacuum. And much of the science that was presented even at today's meeting strikes me as being relevant to understanding broader protection even against other viruses as well. And so global investment in this topic is not going to be wasted. It's going to help us with other pandemic diseases if and when they arise, but it will also help us with other non-pandemic diseases. And so to me, that's all the more reason to keep up the momentum in favor of this work. So with that, I will let you close the meeting, Stanley.
Well, I will just say a few things to close this meeting. And the first is that we must remember the past as well as looking to the future. SARS-1 was a warning and happened about 15 years ago. Now, when will SARS-3 happen? Obviously, we don't know. And it may be 15 years from now, it may be 30 years from now. But if we don't prepare for it, we'll be in the same position as we were when SARS-2 broke out. That is to say, having done little about preparing for SARS-2 after the example of SARS-1. How to qualify such vaccines is an important regulatory issue, which must be solved in advance. That is, we must have criteria for at least showing that a vaccine protects against SARS-1 and SARS-2, and that there are theoretical and animal data suggesting that that such a vaccine would protect against other coronaviruses in animal studies or in vitro studies. I think as a vaccinologist, there's been insufficient use of adjuvants in this field. And I think that's something that should be looked at. I also agree with the suggestion that was made that children could be immunized in such a way that they would have some B-cell and T-cell memory if they are faced with another coronavirus. And I noted that we haven't heard anything about messenger RNA vaccines for the future. Nevertheless, I think if we don't forget, if our memories are sufficient to continue research in this field, we will be prepared for SARS-3. If we don't do that, we will have another disaster. So let us not make the mistake of forgetting the past and think of our descendants as well as our ancestors. And so with that, I think I will close the meeting and thank all the presenters and the audience and WHO. Thank you.
Stanley, thank you very much. You inspired us to start working on this area. You help us today and we will keep going. So thanks to Stanley, to Phil, and to all the speakers and panelists today. We are going to come back. We took note of everything you suggested today, and we are coming back for the next consultation in about 4 to 6 weeks' time. Thank you to all. Bye.
Bye.